We should be. I'm just gonna wait. All right, wow. The conference is finally here after months of planning. I'm thrilled to moderate this session and to begin the conference. Welcome to the 2022 International Virtual GM1 Community Conference. We're so excited for today's event. I'm Christine Wagner. I'm the president and founder of CareGM1. To view the full agenda, you can look at the caregm1.org conference website. There will be breaks. We realize that people can get Zoom fatigue. However, we are pleased to present this virtual format so that families around the world can easily participate in this important meeting. I will keep this brief and then we'll hear from Phil, Jackie, and Holly in this first session. Please note that the GM1 disease overview is for newly diagnosed families, and everyone, the FDA as well, will be part of the externally led patient-focused drug development meeting on Friday, the ELPFDD. And we're so incredibly thankful to the sponsors, all of the speakers, all of the families, and to you all, the attendees, for your participation. Between this conference today and the ELPFDD, there are nearly 50 speakers, so it's going to be jam-packed. Phil's keynote is entitled GM1 gangliosidosis, the therapeutic journey over the next 5 years. Phil has a long history of working in rare diseases and newborn screening. I'm excited to hear from him. Jackie is joining us from Cure SMA. And she's going to talk about learnings from another disease, spinal muscular atrophy, that actually has 3 approved therapies. And I'm hoping we can kind of imagine a future of maybe what a potential could be for GM1. Holly will be presenting the results from the GM1 caregiver preferences study. Many of you participated in that over the past 2 years, and we're excited to share those findings. Next slide. Oh, so there's the overview. Sorry, I'm a little okay, next slide. All right. If you are new to the GM1 community, please allow me to introduce my beautiful daughter, Iris. She has juvenile GM1, also known as type 2B. She was diagnosed at age 5, right before starting kindergarten. A lot has happened. Age 11 was one of the hardest years we ever had. She said her last word, mom, went into her wheelchair, seizures set in, and she received a feeding tube. She's now 14 years old, and GM1 has had a truly devastating impact on her life and on our family. Nonetheless, we do our best to make the most of every moment. However, it is our sincere hope that our work through CareGM1 will be a legacy of hope for her, our family, and hope for all GM1 families. We truly long for the day when families will be able to access a treatment and that the incredible suffering of our community will be reduced. Next slide, please. The sessions coming up today will cover research, clinical trials, and natural histories. We're excited to hear about both preclinical research, clinical research, and the natural histories. Let me just say that the families who participate in natural histories are truly selfless heroes. I want to specifically recognize those who endure all the testing in natural histories to deepen the understanding of GM1. Yet those same people may be ineligible in some cases for clinical trials. New this year, we will have 2 presentations on CRISPR gene editing, and also new is Jillian's talk on enzyme replacement therapy. We're truly thankful to Azapharos, Lysogene, and Passage Biotech, all biotechs presently working on GM1. Next slide. In the afternoon, we'll have a strong focus on families and the family experience. We're excited to have a session this year on sibling support and the impact of GM1 on the entire family. In addition, we're in an unprecedented moment where we can have a panel where families can discuss the clinical trial experience. Another very important talk is on palliative care and how we can manage to live with this horrible disease. Finally, we hope you'll join us for families, the private family connection session at the end of the day to be able to connect with each other, and that will be led by Dr. Al. Next slide. Wow, this year we have a truly special, unprecedented, singular event, the externally led patient-focused drug development meeting, which I mentioned a bit before. This meeting will be to inform FDA and the general public about the true experiences of families impacted by GM1. This event includes live polling, opportunities to submit written comments and comments by phone, and we're really, really looking for your participation. Please, please join us on Friday, and anyone can attend this public meeting free of charge on caregm1.org/PFDD. Next slide. There it is, the link again and the instructions on how to enter the live stream, which you can find on our social media, on the website, etc. Next slide. So yes, this meeting and the ELPFDD would not be possible without the generous support of our incredible sponsors. We're so thankful to them for helping to enable all our important work and for their generous support of the GM1 community. It truly means so much. Next slide. Again, honored and thankful to the speakers for volunteering their time. And I'm thankful to Caitlin from CareGM1 and Jeff and the CareGM1 board of directors, our advisors, and our volunteers. Again, Dr. Al's family connection sessions at the end of the day. I really hope people will join us for that because, we're so spread out all over the world, but the connections and the friendships we form are so important. I want people to know that recordings will be made available after the conference and after the ELPFTD. But yes, the family connection sessions will not be recorded. Next slide. All right, a little bit of housekeeping. I think you've heard this before if you attended our conferences, but just a few disclaimers. Please always consult your doctors regarding medical decisions and clinical trials and medications and all of that. Please remember this is not a confidential forum and it's not HIPAA compliant, and we are recording this and the recordings will be publicly available. Next slide. So this is one of my favorite parts of the conference. I will hand this over to Phil very shortly, but please enjoy this short video of some of the children impacted by GM1 in our community. It absolutely does not represent every person with GM1 or all the sweet angels who passed away. But today and all days, all the children and all the families are in my heart and always on my mind. Living with GM1 is not easy, and losing our loved ones to this devastating disease is a tragedy. But events like this conference truly give me hope for a brighter future. Progress is being made, but time is very much of the essence. So thank you so much, everyone, and we'll play the video and then we'll hear from Phil. Thank you so much. So, Phil.
Thank you very much. Let me just check the audio. Am I fine?
Yep, sounds good.
Okay, great. Good morning, everyone. My name's Phil Riley. Please call me Phil if we have a time for questions later. Christine asked me to begin with a self-introduction, and it's going to be a little bit long because I want to highlight my personal journey in trying to help families who are giving everything they have in their hearts to help children with rare genetic disorders. So I am a 75-year-old physician. I trained in internal medicine decades ago, but early on I got very interested in rare genetic diseases, and for more than 4 decades I've been trying to work with families and developing new tests and new companies to treat these disorders. So I've given my professional life to diseases like GM1. It is so wonderful to see the children's faces, and yet so painful. And I've lived this over and over again, and I can't tell you how deeply I feel that parents are a critical force in moving the therapeutic journey forward. I could cite you over and over again examples of how just a handful or even one person who discovered that his or her little girl or boy was affected with something terrible moved mountains. And the reason I want to mention one story in this regard is that I really strive to offer what I call rational hope. So I modified the word hope. We're all fighting tremendous battles here, and we have to be realistic, but we can be and we should be hopeful. So let me just give you one example about a disorder some of you may or may not know called phenylketonuria. It's a terrible disease that causes severe brain damage. The patients I took care of when I was younger, adult patients, had no language, seizure disorders. They needed total care. It was a very, very hard life for them and their parents, and they lived into middle age often. And then, going way back, about 1930, a mom in Norway had 2 children who were severely mentally retarded, as they said then. And I'll move to the slides in just a second, Christine. She went to doctor after doctor to find out what was wrong with her child. And finally, she met a doctor, a really persistent lady, who did biochemical research and discovered a chemical in the urine of the children that ultimately, 20 years later, led to a diagnosis of what the disease was caused by, a single gene disorder like GM1, and which then led another 10 years later to the understanding if you found out early enough that the child has the disorder and put them on a special diet, you could almost totally avert the disease. That was one of the great triumphs in genetic medicine, one of the great early triumphs. And it gives me hope that we can do other things to help other children with other diseases, no matter how rare or how devastating. So can I have the next slide, please? So I just want to now, I only have about 12 slides, and I'm going to try and lead time for a little bit of discussion. But I want to just say a few words about GM1 because and I know a lot of experts come after me, and I'm not going to steal their thunder. I just want to make a few high-level points about it. The history of our understanding of GM1 is actually very similar to many other rare genetic disorders. For perhaps several decades, typically in the 50s, 60s, 70s, 80s, what doctors did is they reported on the phenotype or the way the disease looked, what its major problems were, loss of vision or whatever it might be. But they were case reports, and they didn't say really anything about the underlying molecular problems. That required technology that we now have, thank goodness, and we can use very effectively. One of the great advances which started in the 1980s, more or less, was enzyme replacement therapy. And I know you have, I think, at least one talk during the conference on that topic. After all, most of these disorders are caused by the inability of some gene to ultimately produce a protein or enzyme that is necessary for normal activity, in this case, in the lysosome. So one of the first great examples of enzyme replacement therapy in the therapeutic world was Genzyme, a company in Boston since acquired by Sanofi, which developed a way to treat certain childhood storage diseases with enzyme replacement. It made a great difference for a number of diseases, but enzyme replacement therapy has been very difficult to use in disorders where a lot of the damage is done to the brain. I mean, after all, imagine how complex the brain is and how many billions of cells it has, and you're trying to get a corrective enzyme into many or most of those cells. So it was helpful, but it was not always successful. Now, as I'll speak about in a few minutes, we have the opportunity to think deeply about gene therapy, a next generation kind of treatment, and then CRISPR therapy you're going to hear about from speakers coming forward. So can I have the next slide, please? So just let me give you a little bit of background about gene therapy history, because I have, first, I've been in the field a long time, and secondly, I think that's likely to be where some of our major breakthroughs are going to be. So gene therapy is only about 25 years old. That may seem like a long time, but it's been slow and difficult to bring gene therapy forward for a lot of reasons, although we're making progress. So in the early days, we didn't know, say back around 2000, we didn't really know how many vectors they're called, what the dosage should be for gene therapy if we were trying to treat something. And also, once you dosed once, it was hard to dose again because the body might have developed antibodies against what was being delivered. Also, about 20 years ago, there was unfortunately a death of a young man in Pennsylvania after he'd been treated with a novel form of gene therapy. And that death set the field back because it was very hard to decide what constituted safety. Another series of events occurred in Paris and London in the period around between 2000 and 2010, where there was some remarkable success with a certain kind of gene therapy, but it was offset by the manner in which the viral vector, the molecule carrying, if you will, the therapeutic agent, integrated into the human genome and cells and actually caused forms of leukemia. So we actually did well by the disease, but caused another disease. So that was a big setback. Next slide, please. So today, we've made immense progress in gene therapy. There are hundreds of academic labs and companies trying to develop gene therapy for many very rare disorders, and some you'll hear about, I'm sure. What are the challenges we face? Well, to make the so-called viral vector that carries the corrective gene to the body and to the billions and billions of cells it needs to reach, we have to make a heck of a lot of it, particularly if it's a muscle disease. And it's still very expensive to make, to construct and manufacture these vectors. We really need to advance our ability to make vectors cheaply and get them delivered. And delivery, we still are really not sure of how much to give. It's by my thinking, how we think about dosage studies is sort of small, medium, and large. There's still a lot to do here, but progress is being made. Distribution is a key question. What do I mean by distribution? Typical gene therapy is delivered into a vessel, just like an IV therapy, sometimes the spinal cord fluid, sometimes fluid around the brain, and it can be in other places, but just because we give it there, we don't always know how well it will be distributed and whether it will be reaching the right tissues. One of the problems is when you deliver in the blood, often as the viral vector passes through the body, a lot of it is soaked up by the liver and doesn't really get distributed the way we would like. The next issue is toxicity. By toxicity, I mean it's possible to give too much, even unintentionally, because of the distribution issue. Sometimes you'll read in a scientific paper that, on average, a cell got 2 copies of the gene. Well, that would be fine, except that it's average. Some cells, many cells may get none, and some cells may get 10 copies of the gene. So it's possible to overdose certain cells. So we're still learning a lot about that. Now, this lack of biomarkers, the last point I'm talking about in this slide, can be extremely important from a regulatory point of view. Work on biomarkers, some either neuroimaging study or looking at a particular chemical in the blood can be immensely important to allow, if it's a strong message of correlation with the disease, to allow the FDA to approve a study early and let it go forward and track it around biomarkers. So one area everybody is listening to me about who is not an expert, you should really learn more about biomarkers and how we develop them. And there's plenty of literature on all these topics on the internet. And most of it is, I think pretty accurate. Next slide, please. So now let's hone in a little bit on GM1 today. It's true. There are, several, the 2 very good ones I know of, and a third I'm aware of. There are several efforts to develop gene therapy for this disorder. So what are the key issues that might slow us down in our effort to reach the ultimate goal of a novel therapy that really will make a difference for our children? Well, one of the key issues is in disorders of an early onset, there's a special problem because the disease has been there in one sense since conception and certainly since birth, although you may not see all the manifestations of it early on. But the longer it progresses, the shorter the treatment window, because it's going to be harder and harder to treat. I think we heard that Christine's daughter was diagnosed at about age 6, I think. For me, that's very late. And I'm going to come back to that later. I've already mentioned brain distribution. I think we're doing much better generally in gene therapy, and that partly because if we think it's best, we can deliver the therapeutic agent, the vector, closer to the brain through the spinal cord fluid or even through something called the cisterna magna at the back of the brain, which is a fairly large empty space, if you will, that neuroradiologists can reach to deliver a therapy. Durability of effect, by that I mean how long will a therapy, even if it's working, last? This has been a very big issue in the last decade around gene therapy for hemophilia, where we get some really fine results, but it seems to tail off after a certain number of years in some studies. So one of the questions is, the holy grail would be to treat once, get the good gene to replace the bad gene, and never have to treat again. But there's no evidence in my mind that we're there yet. I'm not saying we can't get there. By neutralizing antibodies, what I'm referring to is that many of the AAV virus that is used to carry a replacement gene, people have been exposed to that virus, like the common cold, if you will, frequently. And that means the body has made antibodies against it. So even when you're giving AAV, say 2 or 5, that have different names, as a delivery agent, the body might see it as foreign because of prior exposure and sop it up, if you will, limiting the effectiveness of a very expensive treatment. Toxicity and biomarkers we've talked about. Let's go to the next slide. CRISPR. This is a different form of technology that you're going to hear about, and there's great reason to have hope for its application in many genetic diseases. It, instead of replacing a gene, actually has a very fancy biochemical method of identifying a gene in question, a gene with a mutation that you're after, and actually cutting it out so it can be replaced, if you will, cutting out the mutation so that you can revert the gene to a normal state. And a big company, many companies, Vertex is one in Boston that's known for its work with cystic fibrosis, is about to launch a clinical trial with this technology for sickle cell anemia. The big problem with CRISPR, I think still, but it will be solved, is what are called off-target effects. Will the biological material sometimes attack the genes in the wrong place or wrong genes? And I think that's improving, but we're not ready to go, I don't think, for therapy there. Next slide, please. Now, this is a very special topic for me. And if you don't remember anything else about what I say, I really want you to remember my words about newborn screening, NBS. In the United States today, in virtually every state, newborns are given a test. A drop of blood is taken from the heel and sent to a special lab, and about 60 diseases are screened for to see if the baby is born with any of them, including sickle cell, cystic fibrosis, and a number of other single gene disorders. But that's 60 disorders, and there are 6,000 or more single gene disorders that could be identified at birth with different technology. There's great controversy about where we're going to go in the future with newborn screening, but and this is my remarks, many people would disagree. I think newborn screening, given the technology we have, is hopelessly out of date and needs a complete revamping in our society. I don't have the time to go into the structure of the system, but on average, the Federal Advisory Panel that approves, it's not legal, it's just sort of recommendation that some tests be added to a state newborn screening panel, adds about one new disease a year out of these thousands and thousands. Now, why do we say you can make a difference? Well, because one can lobby their state legislatures or Department of Public Health to say, we should screen for this test. There's a screen for this disease. There's a test available. There's a lot that can be done for it. Why aren't we screening for it? And I'm just going to mention two disorders. One, you're going to hear a lot more about SMA, and SCID8 is a very rare immune disorder where kids don't build up a proper defense against infections. In the case of SCID, which was, I think, first adopted as a newborn screening test in California, one woman, a doctor whom I know and I really admire what she's done, Jennifer Puck, and her colleagues in California developed a newborn screening test for this disorder about a decade ago. And now almost every state in the United States screens for this disorder, identifies the rare children early, and there are treatments available for them. So to me, it's an example, two examples, and there will be more, of what parent groups can do by getting together with experts and getting the ear of legislatures. It's not a lot of money to add a test. It is, of course, some money, and state departments of public health don't have a lot of those funds, but that's not a reason not to push it forward. Next slide, please. Just a brief word about carrier screening. What I mean by that is that for all autosomal recessive disorders where the parents are normal, but each one carries a mutation for the same gene, and therefore puts the baby at 1 in 4 risk. It is possible to use carrier screening to alert patients either before the birth of the first child that's affected or after, if their pregnancy is going to be affected. The reason I mentioned the Tay-Sachs success story, this is a rare storage disorder, particularly common in Ashkenazi Jews, that 25, 30 years ago was much more common than it is today, because the Ashkenazi Jewish community, in combination with physicians, did widespread screening within their population and could actually avert and greatly reduce the number of children born with this is a fatal disorder of childhood. And we've seen a reduction in the United States of about 90% or more of children born with Tay-Sachs disease. In the vast majority of cases, that has nothing to do with abortion. It has to do with warning couples about their risks. Next, and this, I think there should be much more carrier screening in the United States for many disorders, not just GM1. And I think with the technology and the lowering cost of gene sequencing, we'll be doing that a lot more in the future. Next slide, please. This is, my title was focused on the next 5 years. I want to give you an update here. This is coming. It's not coming this year, but it's coming. The cost of sequencing of a person or a fetus is dropping like a rocket. It used to be hundreds of thousands. One of the key companies just announced they could offer whole genome sequencing for $200. To me, that's mind-blowing. 25 years ago, it cost $3 billion to sequence the consensus human genome. And the reason I put this up here is when people think about testing fetuses, they rarely think about treatment in utero. But that is also coming. There will come a day, I don't know exactly when, but there will come a day when we'll be able to identify affected fetuses quite sort of in the middle of pregnancy, maybe around week 20, maybe earlier. We can event them, maybe we can identify them very early by 8 weeks, say, by getting a hold of fetal cells, which is already possible, and then possibly treat in the last third of pregnancy. And this has already been done for one rare disorder very successfully. It was published in the New England Journal of Medicine. I was privileged to be part of that. This is called X-linked hypohidrotic ectodermal dysplasia. It's caused by a particular birth defect that could be averted. And we were able to treat 2 little boys in Germany, and they've done very well. So in utero treatment of rare diseases is on the horizon. Next slide, please. Here's a question many parents ask me: why does drug development take so long? Well, it's really concern about safety, and that's the job of the FDA before it addresses efficacy, to look at safety. But I'll say the following things. Some of you have read about Tim Yu and his work with Batten disease in Boston. Tim was able to construct an FDA-approved trial for a single child in the same way, but much more quickly than might be done for a typical study involving hundreds of children. So we call these N-of-1 studies. They're not easy. They're expensive for a family because there's no funding for it for the most part. But the FDA listens to things like this. It's on your side. At my own place where I work in Boston, we are trying to figure out a way to construct a company that will do nothing but N-of-1 studies, where one company develops many different drugs for children with super rare diseases. We're not there yet because it's really tough to make it commercially viable. There are also companies in existence. One of my favorites, and I am affiliated with it, so I should mention that, is Bridge Bio in California, founded by a brilliant young guy named Neil Kumar. Actually has already gotten an approved therapy for molybdenum cofactor deficiency in Europe. It's super, super rare, but despite the fact that his company will never make any money on it, they've got an approved drug for it. So the fact that biotech companies will look at really rare diseases, I think, is encouraging. Next slide, please. How can we accelerate the drug development process? I believe it's you guys who can do that. Patient groups They get the ear of the regulators. They learn from other patient groups. They get top advisors who almost always will consult without charging you. I've already mentioned the push for newborn screening. And what you can particularly do is help on the last two bullets here. I want to know everything about a child's disease from the day he or she is born. And nobody knows that child better than the parent. The knowledge you have about the disease is more valuable than you might realize. It's a matter of collecting it, collating, and studying it. And by the same time, participating in biomarker studies. I really think you guys can make a big difference. Next slide. So, my 5-year crystal ball for GM1. I believe we're going to get, within 5 years, newborn screening for this and a lot of other storage diseases. Many people are working on new and safer and cheaper ways to deliver curative genes through vehicles other than AAV. I think there will be more than 10, perhaps as many as 20 new gene therapy products approved for use in children by 2027. And I would not be surprised if there was a gene therapy approved by 5 years from now for this disorder. CRISPR, I think, needs more time. It's a very powerful therapy, but that implies that we have to be super careful about the dangers of off-target effects. And there will be clinical trials underway for in utero treatment. So I probably, Christine, used up all my time, but I'll stop here and hope that I got some good messages across.
No, thank you so much, Phil. So we'll, since Phil has to leave, we can take 1 or 2 questions, but we're a little bit behind, but we'll take some of the Q&A time.
I'm sorry if I ran too long.
Oh no, thank you. So does anyone feel free to type in the question to the comment if you have something, and we can ask Phil really quickly, and then we'll go on to Jackie. Anyone? Not seeing any questions.
Okay. Well, remember my pitch for newborn screening.
Okay, great. Thank you so much. Thanks,
Yeah.
Phil.
Bye.
And now we'll hand it off to Jackie.
All right. Thanks, Christine, for inviting me to share a little bit about the journey of the SMA community to the 3 approved drugs that we currently have, and share some of the lessons learned. I think there are a lot of parallels between SMA and GM1, so I hope that that sharing the SMA story is informative for the GM1 community. So as I mentioned, currently there are 3 approved drugs for SMA, and about 7 additional investigational drugs in various stages of clinical trials. But what I also want to mention here is the importance of building up the bottom of this pipeline. And that's something that, even though we have approved drugs, they aren't cures. And so I really want to emphasize the importance of continuing to invest in basic research and the preclinical work to really understand kind of more about the pathology of disease. Approved drugs don't necessarily mean that anything is done, and there's always room to learn more and develop additional druggable targets. So these 3 new therapies for SMA that have come about in the last 6 years have really revolutionized and dramatically changed the natural history of SMA. And one thing that we've learned through these clinical trials is that early identification and treatment produces the most robust change in outcomes for patients. And the most striking when these drugs are able to be delivered before the onset of symptoms. We do know that symptomatic treatment is helpful. It's just not as robust as that pre-symptomatic treatment. And we see some slowing of disease, stabilization, and small increases in strength through symptomatic treatment. I'd also like to note that about 40% of SMA patients currently remain untreated for a variety of reasons, but there's still much work to be done in the SMA space. So this is a timeline to these treatments, just to give you a little context of the amount of time that that it took to get here. So the gene that is responsible for SMA, SMN1, and its counterpart SMN2, which we'll discuss here briefly, were discovered in 1995. If you fast forward to 2011, that was when clinical trial programs began, and the first drug for SMA, Spinraza, was approved in late 2016. So you can see the timeline for SMA, and I should note that this is relatively fast. There were several things that were advantageous for SMA, one being the monogenetic nature of the disorder, meaning that there's a single gene that's responsible for causing disease, and that gene was a clear target for drug development. There are over 7,000 rare diseases and fewer than 10% have any treatments. So to have these drugs is truly remarkable for the SMA community. In order to understand the development of these therapies a little better, I want to just kind of give you a brief crash course in SMA genetics. So if you can bear with me for about 2 minutes, I think the remainder of the talk will make more sense. We won't get too far into the weeds, but I want you to kind of understand the basics and how that contributes to SMA type. And so in SMA, you have the SMN1 gene and the SMN2 gene. The SMN2 gene is an inverted and duplicated copy of SMN1, but it's not the same as SMN1. There's a key change in the DNA that causes one of what I'll call a puzzle piece of the gene to be missing in the final product or the protein that's produced from the gene. And so when you have SMA, it's also autosomal recessive like GM1. So patients with SMA are missing both copies of their SMN1 gene, and they're forced to rely on their SMN2 gene. But most of the protein produced from this gene is lacking that critical puzzle piece. So it's producing a truncated, less functional protein, which is the pink here. But about 10% of the time you are getting a full-length, fully functional protein from SMN2. So this truncated protein, while not as functional, is not toxic. And so you can imagine that the more copies of SMN2 a patient has, the more full-length protein they're getting off their SMN2 gene, and the less severe their disease symptoms. And we'll get to more on that in just a second. So what happens when you're missing SMN1? When you're missing SMN1 and you're forced to rely on however many copies of SMN2 you have low levels of SMN protein. And without SMN protein, there is motor neuron dysfunction and death. And when that happens, you get muscular atrophy. And so we see that in varying severities across the varying types of SMA. And so here you can see, as you can imagine, if you think back to that slide, showing SMN2, where the more copies of SMN2 a patient has, the more functional protein they're making and the less severe their disease. And so we see that in what we historically call types of SMA. So type 1 is about 60% of patients, and that's the most severe form. So they have usually about 2 copies of SMN2. Type 3 is an onset around 18 months to 3 years, depending on subtype, and those children generally have 3 copies of SMN2. So a correlation between SMN2 copy number and type and clinical onset and disease severity. And so here, Phil just talked about the importance of natural history, and I want to really emphasize that here as well. Natural history studies for SMA gave us a wealth of information about the disease and have really been pivotal as we moved into clinical trials. And to really understand the natural progression of the disease and how any given investigational drug was able to modify that trajectory. And so here you can see, and I won't dwell on this for too long, but natural history for SMA type 1 infants who historically never achieved head control or the ability to sit was survival of less than 2 years. And that's dramatically changing with the advent of therapies. Now I'd like to talk a little bit about Spinraza, which was the first drug approved for SMA. It's really hard to talk about any of the other drugs without talking about this one, of course, because it was kind of the pivotal initial trials that were done in SMA. And so Spinraza is an IT or intrathecal administration, so directly into the spinal column, and it was approved for all ages and types of SMA. It's you get a bunch of loading doses close together, and then there's maintenance dosing every 4 months as someone remains on the therapy. So backing up to look at how we got to Spinraza, in 2003, Cure SMA made some seed grants to fund Dr. Singh and Androphy for their work in identifying the sequence, which could be modulated in SMN2 to cause that gene to make more fully functional protein. And so then that resulted in a partnership between Cold Spring Harbor and Ionis to further develop this, proceeded through clinical trials, which phase 2 and phase 3 trials were launched in 2013 to 2015, and ultimately resulted in approval in 2016. So this was over 10 years of work from identifying a drug target to approval. And I should note that that is also extremely quick for drug development. And here you can see kind of the timeline of the trials to give you a little context. I won't spend too long here, but just to note that these were multi-year trials, and some of them are still in their open label extension phase gathering data. But I want to talk about the Nurture trial for SMA. And so this was the pre-symptomatic trial. So this was babies born with SMA who were less than 6 weeks of age at their first dose. They had 2 or 3 copies of SMN2, that modifying gene that produces some functional protein, and there were 25 total infants enrolled in this study. And so you can see here, if you'll remember, natural history for SMA type 1 was never achieving head control. And so here you're looking at a crowded graph of motor milestone achievement for these infants. And you can see that they're achieving unprecedented milestones like sitting without support, and even walking alone. And so this was just truly groundbreaking and a whole paradigm shift for the SMA community. Here I'm bringing in the Endure trial, which was for infants less than 6 months at first dose, but were symptomatic. And so you can see, and what I want to highlight here is that the earlier intervention of the Nurture trial, which is the green line and the blue line for 2 and 3 copies of SMN2 respectively, versus down here you can see the line for Endure, and this is looking at motor milestone achievement. So those infants who were treated earlier before their symptom onset had more robust gain of typical motor milestones than those who were treated earlier. They're still doing much better than the sham control, which would be representative of natural history, but not as robust as those treated pre-symptomatically, which leads to my next point, which gets back to newborn screening, as Phil mentioned. And really our slogan kind of became time is motor neurons. In a progressive disorder like SMA, you really have to treat before you lose motor neurons. Once they are gone, it's nearly impossible to get them back. And so we began to think about how to get SMA on the recommended uniform screening panel. We knew we needed a few things. We needed evidence that early treatment was better. We felt we had that with this clinical trial data. We needed an approved therapy. We thought that that was coming really soon. And we needed to be able to handle this difference in symptom onset. So what do we do with a baby who is screened positive for SMA, but has a higher SMN2 copy number, so isn't likely to develop symptoms until later as a teenager or a young adult. And so to handle that, we developed a working group to develop a treatment algorithm on how to handle those situations, because we knew it would be something that the advisory committee would want a plan on how to handle. We were able to submit SMA to the recommended uniform screening panel in March of 2017. It took over a year for them to consider it and add it to the RESP in July of 2018. And then it really, our advocacy efforts couldn't end there. As Phil mentioned, this is just a recommendation to states, it's not a mandate. And so we had to do state advocacy, and families were really critical to that in each state to convince legislators in those states to put money towards this, and that it was important and meaningfully changed the lives of infants who are diagnosed early. And so I am happy to report that here, 4 years out from SMA being added to the RESP, we sit at 98% of newborns in the US being screened for SMA, and 48 states currently screening for SMA. And so we aren't quite there yet, but we're getting very close to having every infant screened for SMA in the US upon birth. And then I want to switch to Zolgensma, which is an SMN1 gene replacement gene therapy for SMA that was approved in May of 2019, and it's currently approved for individuals with SMA who are less than 2 years of age. It's a one-time IV infusion, and it uses the AAV9 viral vector to deliver the SMN1 gene. And here are the various trials of Zolgensma, looking at different ages and stages of patients. These started back around 2014. And I want to focus again on the pre-symptomatic infants. So this was infants who were less than 6 weeks of age and had 2 or 3 copies of SMN2. So these infants were treated with the gene therapy prior to symptom onset. So you can see here the survival data for these infants is really striking. The PNCR here represents a natural history study, and these infants who were treated with gene therapy, we didn't lose any of them. And so you can see just how striking that is with the gene therapy. If we look at infants who are symptomatic at time of dosing, it's not quite as great as infants who are treated pre-symptomatically, but still much, much better than natural history. So really, again, getting at that point that early treatment produces more robust results in highlighting the need for newborn screening. So I want, if I can make this work here, to show you this video here. For the sake of time, I'm just going to kind of move you through. This is a family who had lost their first daughter to SMA at the age of 15 months. Their second daughter was able to receive Zolgensma. And I want to, if I can get up here, I'll show you kind of what get started. We'll skip over this. I really want to show you how she's doing now. I think sometimes a video is worth more than the visual. You can see clearly the difference that gene therapy has made for that girl, for Evelyn. And I would be remiss not to mention the latest approved drug for SMA, which is Evrysdi. For the sake of time, I'm not going to go into a lot of detail on the Evrysdi trials, but it's also a splicing modulator. So it causes that SMN2 gene to make more fully functional protein. And it was approved in August of 2020, initially for individuals older than 2 months, but since then it's been approved for infants less than 2 months as well. And it is a liquid medication that's given daily, orally, or by G-tube, and we see remarkable results from it as well, especially again when given before the onset of symptoms. And so where are we? We know that these current therapeutics, while they produce dramatic results, are not cures. Even children who are treated before onset of symptoms are not free from some of the symptoms of SMA. We know that these current therapeutics slow or even stabilize disease regression, but unmet needs remain, even across the disease spectrum, even in those pre-symptomatically treated infants, but especially for adults with SMA for whom treatment wasn't available when they were younger. And so we really think that we need a combinatorial approach to treat SMA. So these drugs that I discussed are SM independent in that they focus on the genes missing or impacted by SMA. And what we're working towards now, and a lot of the clinical trials are focused on, are SMN independent pathways. So thinking of ways to combine drugs to produce more robust therapeutic benefit. And so I want to just bring out some kind of the key lessons that we've learned, which is one, that having an approved drug doesn't mean that the work is done. And like I said earlier, we really are focused on continued investment into basic research to understand some key questions we don't yet know, like how low levels of SMN cause motor neuron death. We know they do, but we don't understand all the mechanisms there. And really to be able to identify other druggable targets. We know that these current therapeutics work best before symptom onset, and so that leaves large unmet needs for individuals who were not treated before onset of symptoms. And the need for biomarkers for SMA would really help us to understand treatment benefit and help us know when to initiate treatment for those rarer cases of later onset SMA, knowing that we need treatment to begin before disease progression starts and motor neurons start to become dysfunctional or die. It also highlights the importance of newborn screening and the amount of advocacy and technical support that goes into that, helping states, public health labs understand the assay, understand how to handle a positive screen, making sure the infrastructure is in place for that, and really the advocacy of families who shared their stories with legislators, making funding decisions for SMA newborn screening, deciding whether to implement the condition about the positive impact that newborn screening had made in their lives. And then continued engagement with the FDA about unmet need. I know that the GM1 community is gearing up for their PFDD meeting, and that's wonderful. I think the FDA is really receptive to patient stories, and Cure SMA has been fortunate to hold a PFDD meeting. We recently held a patient-led listening session emphasizing the remaining unmet needs in SMA and the continued need for investment into drug development for SMA. So with that, I'll stop there. Thank you, Christine, and I can take questions now or later.
So let's just do these one quickly, and then we'll go into Holly. But one is, how are you able to get families in the SMA community to help support and push through clinical trials and FDA? Do you have any recommendations on ways for GM1 families to get involved in moving treatments forward?
Can you repeat the first part of that? Sorry.
How are you able to get the families in the SMA community to help support and push through clinical trials?
Yeah, so the SMA community was very willing to participate in clinical trials, especially initially. And so there was a lot of advocacy being done with FDA. I didn't go into a lot of the details for the sake of time, but a lot of these early drugs were awarded fast track and other designations through advocacy of the community. In their FDA interactions, highlighting the need for treatment. The robust natural history studies that families willingly participated also enabled us to have comparative data in these trials that helped us gain momentum and move forward with trials. So I think there was just a really good stage that was set with FDA engagement, families engaging and sharing their stories, and then robust natural history to kind of support these trials as they launched.
Okay, great, thank you. And then it says, what is the greatest barrier to treatment? Have any SMA patients received gene therapy twice? So that would be interesting. Do people get Zolgensma and Nuzirin, or however you pronounce it?
Yeah, so this idea of combining these drugs is really kind of where we're at in the community. And so there are various clinical trials looking at adding these drugs on top of each other. There are a lot of people who are just doing it in the real world. Access becomes a bit of an issue in that it's sometimes hard to get payers to pay for more than one, but a lot of families received one of the drugs in a trial. And so they are able now to have private payer access to a second drug. And so we are collecting a lot of this data through our patient registry to try to get a handle on what's happening in the real world, how families are accessing these drugs, how they're combining them, and that sort of thing. And there are trials now looking at different combinations of these drugs to see if combining them can produce better results for patients. Getting at the redosing of AAV, no, that has not been done for the antibody reason. So at birth, if a family elects to put their child on Zolgensma, or I shouldn't say at birth, if a family who has a child less than 2 years old who would be eligible for the gene therapy Zolgensma wishes to pursue that, that child is screened for AAV9 antibodies. And so if there are, they cannot receive Zolgensma if their AAV9 antibody titer is too high, because there's risk of a massive immune response. And so redosing with AAV9 is currently not under consideration because once dosed, you would produce antibodies to it. And so redosing would trigger a massive immune response that could be seriously detrimental to the patient. So at this time, no. However, patients who have been treated with gene therapy have gone on to take the other splicing modulators. So those drugs that try to modulate SMN2 to produce more SMN protein instead of replacing SMN1. So a little different angle with those, and they can be combined.
Great, thank you. I think we're out of time. I'm not sure if you're sticking around for the Q&A after Holly's talk, but let's, I'm really pleased that Holly from RTI will be presenting the study on caregiver preferences. Thank you. Thank you so much.
Thank you. Can you hear me well?
Yes.
Great. Okay. So I'm Holly PA. I'm at RTI International. And we've had the pleasure of working with Christine and the GM1 group for the past year and a half to do a study to characterize caregivers' assessments of symptom impact and most important symptom to treat. I'm not going to talk to you about newborn screening, but I'm going to just really quickly, like everyone else, make a plug. Since newborn screening keeps coming up, I completely agree about the importance of newborn screening. And I think it's something that this group should continue to think through ways to partner and think about opportunities to run a newborn screening pilot. We were very lucky in North Carolina to actually work with Jackie and her colleagues and to do a study prior to SMA going on the RUSP that generated some evidence, but also that importantly allowed our state, North Carolina, to pick SMA up very quickly. North Carolina is not known for picking things up as quickly as some other states because of the way that approvals happen in different states. So I will just reiterate the importance of newborn screening down the road for successful treatment of children early. And with that, I'm going to go ahead and talk about what I came here to talk about, which is not newborn screening, but the study that we conducted in conjunction with the community. Am I able to advance these slides?
Oh, no, you just say next slide.
Okay, next slide, please. All right, fantastic. If I didn't already mention this, it's because I have COVID fog, but I'm actually recovering from COVID and I'm having a little bit of COVID fog. So I apologize if I'm not on top of my game right now. But I'm going to tell you a bit about the study that we conducted. It's in 2 phases. So I'm going to talk about the objective, the participants, go through a bit about how each of the phases was set up, and then talk to you about the results and the kind of conclusions from the study. If we did our job right, many of the parents who are involved in this meeting will nod and say, yep, that sounds right. And if you have additional feedback after you hear this about ways that your experience differs or other thoughts and comments, I think actually I would love to hear them. Christine would probably like to hear them too, and it actually might be interesting fodder for continued engagement with the FDA. So our objective was to describe unmet treatment needs in children with GM1, and then to identify among those priority symptoms to treat. So talking about the context of a non-curative treatment that would not take care of all symptoms simultaneously, what would caregivers' priorities be? And then as well, we described the burden and impact of GM1. And this is not a formal impact study. There's no economic analysis. This is a qualitative exploration of burden and impact. The participants in this study are caregivers, and in our case, this all happen to be parents of children with GM1 types 1, 2A, and 2B. Parents who participated had to have a child diagnosed at least 3 months prior to participation. We did not want to get people right when they had a diagnosis because we wanted to learn about their experience having assimilated a diagnosis. And we also allowed bereaved parents to participate if the child had died within the past 3 years. As I mentioned before, the study was sponsored and led by the Cure GM1 Foundation. It was a fantastic experience with us working with Christine and her colleagues with collaboration from NTSAD. And the study had a multidisciplinary advisory committee, and that included academic individuals, people from industry backgrounds, and parents. And so I actually flipped through the attendee list and I see a few of you in the meeting. So I am again grateful for your participation in the study. Next slide, please. So as I mentioned before, we had 2 phases of our study. The first phase was a brief survey to characterize the participant and their children, followed by focus groups. So we did 3 focus groups, and we had 3 to 5 parents per group, and the groups were actually separated based on whether the children had type 1, 2A, or 2B. We conducted those in the summer of 2021, and those were all conducted in English, although participants were from many countries. And our aim there, again, was really to focus on burden and start to explore the features and symptoms with the greatest impact. And we asked participants, respondents who were parents in this study, to separate impact on the child and impact on the other people in the family. In the second phase, we intentionally built upon the first phase, where we used the data that we had obtained during the focus group to develop a more nuanced survey that included a quantitative preference weighting, a prioritization activity, where we asked parents to indicate most and least important symptoms to treat from a list that we developed from the focus groups. And then we actually discussed what they had selected during their one-on-one interviews. Those were conducted in December through March 2022, and we actually conducted those in either English or Portuguese. And again, we had participants from many different countries, including families in Brazil for the interviews conducted in Portuguese. And our aim is similar. Again, this was the next step. So here we really were trying to drill down on the most important and impactful symptoms to treat. Next slide, please. So this is a bit of a busy slide, and I'll just highlight a few things. So first of all, we had participants who, it's always nice when we get dads to participate. So thank you, dads. It's sometimes a little bit harder than getting moms to participate. So we were pleased to have dads and moms participate in both the focus group and the interviews. You can see the information we have about race, and we were also pleased, that we were able to have some individuals involved in the study who were non-white, and many who, excuse me, individuals who reported having Hispanic or Latino background. That's, again, always a nice thing for a study like this, although a qualitative study like we've developed is not intended to be generalizable. You can see, maybe more importantly to the data, I'm going to describe what types. So we had three early infantile types in the focus groups, 6 2A and 4 2B. And in the interviews, you see we had very even groups with 9, 9, and 8. So next slide, please. Sorry. Okay. So in the focus groups, these were the symptoms that actually came out, and they were generated by the parents in the focus groups. And so we've combined the 2A and 2B, and I'm sorry, the B's missing there, into a single group because they were quite similar. So I'm going to start on the type 1 list. So there was a lot of symptoms description around communication and awareness and engagement with caregivers. There were challenges with muscle tone, both hypotonia and hypertonia, basic gross motor skills, vision and hearing, swallowing and feeding, seizures, frequent illness, and pain. And in type 2A and 2B, you see some overlap, although there's a bit of nuance there. So we, again, have a lot of concern about communication, more gross motor skills. Again, not surprisingly, those gross motor skills were a bit higher level because the children had attained higher milestones. Similar concerns about muscle tone, fine motor skills, swallowing and feeding, seizures, sleep, toileting, pain, and then generally regression. So parents talked about the kind of challenge of having skills that were lost. So it's not a specific symptom, but that general concept of losing skills. And I'm going to go into more detail on these, so you'll see a bit more information. Next slide, please. So you remember then that what we did was we took those lists that were generated from the focus group, and we actually asked parents to rank them using a prioritization activity called best worst scaling. And so the symptoms here are for type 1. These were the 9 parents who reported about their child with type 1. And you see the items that are in this list are actually in order. The items will look familiar because they're, again, generated from the focus groups. And the one that's added here is lifespan, because many parents talked, of course, about the fact that their children were likely to die soon.
Uh-oh. Did we lose Holly? Oops, she's going to be coming back.
Oh, weirdly, I lost my connection. So can you hear me again?
Yes.
Okay, that was very strange because I didn't get any warning about that. Anyway, what I was saying was that the important thing to understand about this list is that when we talk to parents about these later, and again, I'm going to show you some of the qualitative data in a minute, parents really had a hard time putting these things in any kind of priority order because they were all very important. And one of the things we anticipated that we did indeed find was that many of these symptoms that were important at one stage of illness became less important later, or vice versa. And so there was some moving around of items on this list and a lot of qualifications for how parents prioritize them, which I'm going to show you in a minute. So the next slide shows the GM1 type 2A and 2B. You see the items are in reasonably similar order here, which is pretty much what we expected. And again, the communication and lifespan are around the top and getting around mobility. So we're seeing some sort of similarities in these groups about what things parents are prioritizing. And I'm going to go ahead and have you move to the next slide, because now I've lost a bit of time by being knocked off of Zoom. And I'm going to show you a bit around actually what parents told us about why these things were important and how they characterize them. So next slide, please. So again, I mentioned this before, that parents, really didn't like having to prioritize these, and it led to really great discussion. And that was our whole goal. And so one of the things that came up was that the items that we gave on the slides before were considered by parents to be very interrelated, which is not surprising. And so things were anticipated to sort of move together. And the other thing is that parents generated sort of umbrellas of symptoms under which they felt like there was meaningful prioritization. And so those buckets that the parents came up with are in these topics here. So it's interaction and engagement, lifespan and quality of life, which are together for a reason I'll discuss in a second, muscle function and tone, eating, feeding, and pain. So next slide. I'll give you a moment to read this while I drink my tea. Okay. I know you're probably not done, but so the interaction and engagement component is, as you would imagine, very important to families. And families talked about this in a number of different ways. Some of them are relatively straightforward, like the first comment here about he can't tell us what is going on, and sometimes that makes it harder. So there's a lot of challenge in the interaction and communication, or being able to meet the child's needs. There were also many parents who talked about, so caregiving, like we really want to be able to caregive well for our children, and this interaction component is difficult. There were also many, many challenges related to the child's ability to express themselves in a meaningful way. And you see that in the second quote from a parent who has a child with type 2A. So, just reduced ability to react in ways that are apparent to the caregivers, and the association in the caregiver's mind that may be related to less sort of engagement and enthusiasm, and again, sort of general happiness and quality of life. The last quote I think is really interesting, because it also shows that the importance of a reciprocal relationship, and how important engagement is in interaction are to the ability of the caregiver and the child to maintain that important connection, which is both important for both of them psychosocially, but also really helps the caregiver be able to do the best care and stay engaged. And so I think that parents mentioned many different ways that this overarching concept is really important. Next slide. We found a lot of data in the studies. I'm going very quickly. I apologize for that. In terms of lifespan and quality of life, as you saw in the prioritization, lifespan was very important to parents. And there was something that, we heard parents talk about quite a lot, but most parents were very clear that lifespan was only important as long as it was associated with a good quality of life. And at the time in which quality of life was reduced, that lifespan would no longer be a valued component. And so you see on this slide a range of different perspectives from parents going from type 1 to type 2B about how they value lifespan. And the first quote is really around the idea that parents don't begrudge their children the care they're giving them, and that this is actually a value to the parents. And so this was an important thing that came through with all of the data we collected. But the second two quotes really indicate the importance, like I said, of that balance between lifespan and quality of life, where the second quote is talking about, they want the child to live as long as he can, as long as he's happy and as long as he's not in pain. And then if he's in pain or he's not thriving, then parents would make a different decision about the importance of that element. And the third is from a parent whose child was further progressed and, felt at this point that the child was, potentially having too much pain and suffering for the lifespan to be meaningful anymore. So we saw that full range. Next slide, please. So as I mentioned before, muscle function and tone were also very important. We saw them being important in sort of different ways in types 1, 2A, and 2B. Muscle function and tone was related to ability to, get around and have some independence, but also it was discussed by parents as being very important for general overall health. So, the children, if they have tone issues, may have more pain, may have less ability to move themselves in ways that prevent them from having issues with skin breakdown, would cause frustration for the children. And so you can see, again, a range of different quotes here from parents going from type 1 to type 2B about the potential implications of muscle function and tone, from pain in type 1 to frustration in type 2A, and to this ability of maintaining independence and how important that some degree of independence is in type 2B. Next slide, please. So the next two slides I have less, no quotes, less information, because these were, while again, very important, were considered to be less overall important than the first three domains. Eating and feeding is, of course, important to growth and survival, but the thing that came out really importantly as well is that parents talked about the importance of eating and feeding being enjoyable, and the challenge to the enjoyment and the social interaction and the sort of overall, again, quality of life component that comes with eating that had been challenged by GM1. And so that was something that parents talked quite a bit about, and ways to continue to allow the children to be as involved as possible in eating and in family and social environments that involved eating was very important. Next slide. And then pain ran throughout. You saw indications in prior quotes about pain. Parents were very motivated to avoid pain or the potential for pain in their children, as this was perceived to be a primary threat to quality of life. And one of the challenges that parents talked about is whether they would know that their children were in pain, and would there be a time in the illness progression where their children may be uncomfortable or in pain, and they would not know that and thus would not be able to intervene. And that caused parents a lot of distress. Next slide. Okay, this is the last result slide. So we, as again, this is not going to be surprising to you, we did find that there was significant burden of GM1 on both the child and the caregiver's lives. Parents talked about the time-consuming and complex care that's required, the lack of independence and really significant dependence on caregivers, and children's frustration and sadness as symptoms progressed. The need for lifestyle adaptation that reduced parents' ability to have employment, leisure, and travel options, and challenges to social support, and the really significant ongoing emotional burden that came with coping with diagnosis and then anticipating symptom exacerbation and end of life, with a lot of uncertainty that many parents found to be very difficult to manage. And yet overall, parents demonstrated really remarkable adaptation, and most perceived their children to be happy. So I know we talk so much about the negatives in these studies, characterizing things that are negative, but I do want to reinforce that parents talked a lot about the benefits that having a child with GM1 had brought to themselves and to their families, and how they were just really showed remarkable adaptation. So next slide, and I will wrap up. So this is the first study of experiences, perspectives of caregivers caring for children with GM1. The quality of life is really paramount in parents' prioritization of symptoms to treat. They, again, every decision was related to that ability to maintain as high a quality of life as possible for as long as possible. And although lifespan was highly valued, most parents would not want to extend life without having an acceptable quality of life for their child. We developed this study primarily to inform future drug development and to inform things like outcome measures that could be looked at for future clinical trials. And so we also have as you saw, a very concrete list of symptoms and the way parents characterize those symptoms that we think will also really be informative for advancing drug development. And with that, next slide, I will thank my colleagues who participated in this study, and this is the authors listed here. And again, appreciate the support from GM1 and their supporters so that we could actually do this study. So thank you very much. Happy to take any questions.
Great. Thank you so much, Holly, and everyone for this this great session. So we have a question, and Jackie, if you want to come back on, feel free. From Joe, it says, is the FDA sensitive to risks benefits of rare disease clinical trials in your opinion? There may be side effects, Zolgensma, but do they consider that the benefits are worth the risk?
Yeah, so yes, I think FDA is considerate of benefit risk. And one thing that the SMA community did was back in 2017, kind of as a follow-up to our PDFDD meeting there, we conducted a benefit risk survey. So just collecting input from members of the SMA community, whether they be affected individuals or caregivers of affected individuals, to really get a handle on tolerance for risk for given benefits. And so we were able to kind of quantify that information and share that with FDA. And we found that families were willing to undertake considerable risk for the chance at benefits. And so that information was shared with FDA and through other advocate testimonies from with FDA. I think they had a really good sense that the SMA community was willing to tolerate considerable risk. And we are currently in the process also of updating that benefit risk survey to see kind of how risk tolerance has changed with approved therapies, if it has. And so that's kind of an ongoing dialogue with FDA, with our community, to ensure that they're understanding the risk tolerance of the community as well as their unmet needs.
Yeah, I agree. We've actually done lots of studies about benefit risk and how people weigh that across different especially rare disease areas, and the FDA is quite receptive. And in fact, they have a whole set of guidances around how they want that kind of studies, those sorts of studies to be conducted. And it's much more straightforward than it used to be to do that kind of work and know that it's going to have the maximum impact. One of the challenges that your community would likely face, it's not, we do a lot of studies like this, but I will say it is a challenge, and Jackie, you probably experience this, is when you're talking about hypothetical situation. So we can imagine what a gene therapy, for example, will look like and what kind of risks will come with a gene therapy. But then, once you actually get further in drug development, like Jackie said, we often need to do another study because people's tolerance changes over time. And as we learn more about particular therapies, a lot of times the studies become increasingly targeted to a specific therapy, whereas we generally start more broadly looking at willingness to accept kind of broad constructs. So risk of death, risk of long-term disability, additional disability, things like that. But the FDA is receptive. There's also a question for me in the chat. Should I answer that, Christine?
Oh, yes, sure.
Okay.
Do you have any information on when this important work will be published and where?
Well, we never know the answer to that question. It has been submitted to the American Journal of Medical Genetics, and we've actually responded to review. So it's still under review, but we've received a revise and resubmit, which is always very positive. And so we're hopeful that we will have information about that very soon.
Great. And then we have a question, I think for Holly, on what sorts of sleep issues were discussed as impactful symptoms? Was it more on the side of sleeping too much or disrupted sleep?
Yeah, that's a good question. It was more on disrupted sleep, but parents were very clear about that. And that's why sleep kind of went to the bottom, is that sleep, for most of the parents, they describe sleep as being a problem more for them than it was for the child. There were some cases when parents talked about some challenges for the child not feeling well because they didn't get enough sleep, but mostly it was an issue that was a caregiver issue, and caregivers minimized, you all are amazing, and caregivers minimized their impact for the child's. So that's why sleep went lower on the list.
Okay, great. And then there's just a nice compliment for Jackie that someone really enjoyed the SMA presentation. So very helpful overview, Jackie, nice job. And nice job to everyone. And if anyone has questions for Phil that they would like to email even after the fact, info@caregm1.org, or I'm happy to send them to him and field that. But otherwise, we will have a break if there are no more questions. All right. All right, so the break, we are supposed to be back here in about 17 minutes. So, so please come back for the next session, which will be on the preclinical research, natural histories, and clinical trials. So thank you, everybody. Thank you, Holly, Jackie, and Phil, for that awesome opening.
Thank you.
Oh, one from Michael Gelb. Oh, okay. All right. Mike, I will mail that to Phil. All right, thanks. Okay, see everyone back at 10:50 a.m. or 1:50 or sorry, I'm mixing up the times. Yeah. Yeah, that's Pacific time versus Eastern time. So thanks, everyone.
Hi, Jeff. I think we are about ready to begin the next session. Can everybody hear me okay? Yep.
Yep.
Okay, great. So thanks everyone for your participation so far and for the presentations earlier this morning. I'm super excited to be moderating this panel, which is about all of the steps that scientists, clinicians, and industry are making towards finding a treatment and a cure for GM1. So we have several talks that are planned for presentation about ongoing research for treatments for GM1, as well as clinical trials. And importantly, we'll have a talk on the natural history study that is being conducted for GM1, which is critical for any company who's developing a therapeutic for GM1. So with that, I will pause and hand it over to our first speaker. Just a quick note, we're gonna have each presenter give their talk, and then at the end of the session, we'll have time for Q&A. And also, I'll just quickly ask each presenter to introduce themselves and their affiliation. And so with that, I'll hand it over to Dr. Ra.
Thank you. Can everyone see my screen? Is it there?
Yep.
Yes.
All right, perfect. Good morning, everyone. I'm honored to be here with you this morning to share about genome editing and briefly our lab's work examining the efficacy of genome editing for therapeutic application in GM1 gangliosidosis and other rare pediatric diseases. In the US, a rare disease is defined as a condition affecting less than 200,000 people. Rare diseases affect at least 1 in 16, or approximately 6.2% of the population. And of those affected, about 70% are pediatric and 40% have genetic etiology, with the number of genes that map to rare diseases growing. A subgroup of rare diseases, lysosomal storage diseases or lysosomal diseases, are rare inherited metabolic disorders. As you all know, there is no cure for LSDs, and many do not yet have specific treatments. LSDs result from the defect or loss of a lysosomal protein and lead to lysosomal storage of its substrate, cellular dysfunction, and gross pathology. And unfortunately, these diseases are life-limiting and often terminal in childhood. With more than 70 distinct LSDs and a combined prevalence of about 1 in 5,000, individual lysosomal storage diseases may be rare, but as a group, their frequency is similar to hemophilia. For LSDs, few FDA-approved therapies exist. Clinical trials are ongoing, examining the efficacy of gene therapy, enzyme replacement, substrate reduction, and others for the treatment of LSDs, but the majority of care is focused on improving or maintaining quality of life. Clinical trial data and patient outcomes from FDA-approved therapies indicate that there are unmet needs, and treatments do not fully attenuate clinical symptoms. So challenges exist when modulating the immune response, and also when we're looking at treatments that require recurrence. Current research is focused on defining disease mechanisms and developing efficacious therapeutics, with core areas of interest including enzyme replacement, enzyme enhancement, substrate reduction, stem cell transplantation, gene therapy, and more recently, genome editing. For some LSDs, lifelong treatments with recurrent dosing include enzyme replacement and substrate reduction therapy. But these two types of therapy do not fully attenuate clinical symptoms, so you're still having some effects of the disease, and they don't often reach sanctuary tissues like the brain in the case of enzyme replacement therapy, or may have adverse side effects like substrate reduction therapy. Enzyme enhancement therapy or chaperone therapy is an interesting approach, but it relies on the presence of residual full-length or truncated protein with specific alterations. So this approach actually fails to address all disease-causing mutations and would only be available to a subset of patients if successful. For single dose treatments meant to cure the patient of the disease, like gene therapy, which represents a promising approach, or cell replacement therapy or genome editing. In the case of gene therapy, which relies today on AAV, and the introduction of genetic material via episomal expression is diluted by cell division in actively divided tissues like the liver, which is where a lot of gene therapy is targeted. For GM1, when it's targeted to the brain, as long as it is able to access neurons, it should be sustained over time. For cell replacement therapy or stem cell transplantation, it's a viable option for a few LSDs. However, these one-time treatments do not fully attenuate clinical symptoms in human patients, and there's a significant risk of morbidity and mortality. Genome editing has not yet been translated to the clinic for LSDs. However, it remains a promising avenue of exploration. Genome editing targets and corrects the underlying mutation, which reduces disease burden from the mutant protein. Recent studies have shown that limited immunogenicity exists for genome editing cargo when delivered via lipid nanoparticles, allowing for repeat dosing if or when necessary. With recent advances in genome editing, the development of these curative treatments will continue to expand over the coming years. So what is genome editing? We've heard a lot about it even today. Let's get into sort of what the mechanism looks like for that. And so genome editing is a method that is used to therapeutically target genetic diseases at the root cause. So it's targeting the gene that's affected. This looks like changing the pathogenic or disease-causing mutation variant in the genomic DNA to a non-pathogenic or non-disease-causing alternative. There are many different types of genome editing, but today I will go through some of the advances in CRISPR-Cas-based strategies that have led to improvements in therapeutic efficacy. CRISPR-Cas-based methods rely on two main components: a guide RNA, which is a small strand of RNA that is exactly complementary to the target site. For newer CRISPR-Cas strategies, that means it is specific to the patient's mutation, and each strategy can only recognize and correct the specified mutation. The other component is the Cas nuclease. These are essentially molecular scissors, which are guided to the target site by the guide RNA, where they cut the genomic DNA to encourage cellular DNA repair and the incorporation of specified edits. So you can see here in the first panel, the cutting of that genomic site. So if a child with GM1 has an A where they should have a G in their GLB1 gene, CRISPR-Cas will change the A to a G, and the cellular repair mechanisms will help facilitate that installation. So what does that look like visually? The Cas nuclease forms a ribonucleoprotein complex with the guide RNA. So this sort of blob here is supposed to represent that Cas nuclease, which is a protein that we have engineered in the lab from bacteria. And so it's forming a complex with our guide RNA here. This complex then targets the complementary genomic site in the nucleus. And so this next piece of nucleic acid represents your genomic site. So the guide RNA is binding the nucleotides at your genomic site. Upon binding, the Cas nuclease then cuts both strands of the DNA. When CRISPR-Cas-based genome editing strategies were first studied in the lab, cut DNA would be repaired via two mechanisms: non-homologous end joining, which results in random deletions or insertions of DNA at the cut site, or homology-directed repair, which relies on the incorporation of a donor DNA harboring the specified edit. Both of these strategies are error-prone and have little utility in delivery to most patients. In the past 6 years, advances improved how we utilize CRISPR-Cas in targeting correcting specific mutations to target single nucleotide changes of like nucleotides, so nucleotides that structurally look similar to each other, tethering a secondary enzyme to the Cas nuclease has led to a 90% editing efficiency at many sites. So this addition of a deaminase actually facilitates the conversion of, in this case, an adenine or an A to a guanine or a G. So this one goes from A to G. Prime editing, a second strategy, emerged as a CRISPR-Cas mediated strategy for addressing nearly all mutations, including single nucleotide changes of like nucleotides. In this strategy, a reverse transcriptase, so this little green blob up here is another enzyme attached to the Cas nuclease, is tethered to the Cas nuclease, which is then used to encode new genomic DNA with specific changes at the target site. With these two strategies, it is now possible to make precise edits correcting nearly all mutations, with an estimated 89% of all known pathogenic mutations within the human genome. In our lab, we have focused on the use of base and prime editing for patient-specific alterations in several diseases, including GM1 gangliosidosis, and we're using these to improve therapeutic efficacy at specific sites. So what does genome editing look like in the clinic? We already know that genome editing has potential, and in the past 8 years, we have seen its efficacy translated to the clinic. The world's first in vivo CRISPR-Cas therapy to treat Leber congenital amaurosis in the Brilliance clinical trial via subretinal injection. Treatments for sickle cell anemia and transfusion-dependent beta thalassemia via ex vivo editing of patient stem cells. So that's essentially when we take patient stem cells from the patient, edit them, and then put them back in. And then in vivo delivery via lipid nanoparticles of CRISPR-Cas for the treatment of hereditary transthyretin amyloidosis and hereditary angioedema. We've also seen application in cancer with CAR T-cell therapies and the treatment of chronic infections. This summer, we saw the first patient treated with base editing in vivo, so via intravenous injection with familial hypercholesterolemia, excuse me. These therapies have had few adverse events and paved the way for future, more personalized genome editing strategies. So what does this look like for GM1? We know that GM1 is a rare lysosomal storage disease of autosomal recessive inheritance with an incidence of about 1 in 100,000 to 1 in 200,000. It principally impacts the central nervous system with progressive neurodegeneration and results from mutations in the GLB1 gene. GLB1 encodes the lysosomal enzyme beta-galactosidase, which is responsible for removing the terminal beta-D-galactose residue from GM1 ganglioside during the first step of degradation. Without removal of this residue, GM1 ganglioside and other glycosaminoglycans accumulate in the lysosome and cells, especially neurons, and cause cellular dysfunction. The genetics of GM1 are important for understanding how CRISPR-Cas-based strategies might be efficacious. The GLB1 gene is composed of 16 exons, which are numbered here, with pathogenic variants identified throughout. In a brief analysis of about 200 variants, we identified 45% that are targetable by base editing, and nearly all are targetable by prime editing. With the number of different pathogenic variants, CRISPR-Cas-based approaches represent a truly personalized approach to medicine. We are currently using base and prime editing to correct several mutations in the GLB1 gene, and I'm going to share some brief data from an in vitro or in-cell study examining the efficacy of base editing in a compound heterozygous patient line. So this is just a couple slides of data that we have on a specific CRISPR-Cas project. Cells for this project were obtained from the Coriell Institute and are dermal fibroblasts or skin cells. The cells were obtained via biopsy from a 7-month-old female patient with infantile GM1. The patient is compound heterozygous, meaning they have 2 different mutations in separate genes causing GM1. The base editing site C380 G to A, 380 being the position of the nucleotide in the gene, with A being the pathogenic nucleotide and G being the non-pathogenic or unaffected nucleotide, was targeted using an adenine base editor. This editor converts the adenine to an intermediate that after cellular DNA repair is converted to a guanine. The patient's cells have an average beta-galactosidase activity of 0.6% of normal levels. And on the right, you can see 3 sequencing files where each bump or peak represents a different nucleotide in the GLB1 gene. In the top panel is an unaffected individual with a non-pathogenic G highlighted. The middle panel is our compound heterozygous patient with both the A and G present. The middle panel is highlighting both A and G because the G or non-pathogenic nucleotide is in the GLB1 gene with the other mutation, in this case, the 481T to G, so it looks non-pathogenic at that site. The bottom panel shows the sequencing from cells that have been treated with base editing. There is a significant increase in the amount of the non-pathogenic G nucleotide at this target site. We followed the study over time out to 27 days and determined that the editing at the target site is sustained. You can see that the frequency of the non-pathogenic nucleotide is maintained around 70 to 75%, which is an increase from its 50% presence in the compound heterozygote. This editing was sufficient to yield a consistent increase in the beta-galactosidase activity over time, showing that the editing of the GLB1 gene using CRISPR-Cas base editing was able to restore beta-galactosidase activity. We further characterized the disease pathology in these cells using an assay to detect GM1 ganglioside content. So basically, how much GM1 ganglioside is accumulating in these cells before and after base editing. And in unaffected cells, so these are cells from a patient without GM1 gangliosidosis, we see very little GM1 ganglioside. In the cells before base editing, we see a lot of GM1 ganglioside accumulating. And then in the cells at 22 days after base editing, we see that most of the cells have significantly reduced levels of GM1 ganglioside. I'm quite a bit over time here, so I'll just quickly go through the summary here that CRISPR-Cas-based genome editing is therapeutic. Prime and base editing can target pathogenic mutations in GLB1, and we've shown that base editing can efficiently target the GLB1 gene to correct the 380G to A pathogenic variant, and that it restores beta-galactosidase function and results in lower GM1 ganglioside content. So with that, I'll thank my lab here, the Wong Lab at Chalk Children's, and our funding sources.
Thank you so much, Alessandra. Thanks for the tutorial on CRISPR gene editing and for sharing some of your data. And now I'd like to turn it over to Dr. Prisbilla of the University of Minnesota.
I want to make sure everybody can hear me.
Yep, you're good.
Okay, perfect. So hi, everybody. My name is Michael Prisbilla, and I'm an assistant professor here at the University of Minnesota. And I want to thank Christine specifically for inviting me to give this talk to you guys today. And the title of the talk is going to be Employing the PS System to Correct, excuse me, oh, I lost my screen. Employing the PS System to Correct Beta-galactosidase Expression in GM1 gangliosidosis. Next slide, please. So I don't know why I have the watermark from Chalk on here, but I'm going to move forward with my slides. In the next few slides, I just want to give a brief background on what I work on, the work that I'm going to be discussing today. And so because Dr. Ra had such an incredible introduction to gene editing, CRISPR-Cas9 based genome editing, I'll come over a few parts. And so our group has been working on a platform technology that can be utilized to treat most lysosomal disorders, which we're terming the PS gene editing system, or PS system for short. So starting with the left side of this slide, we have designed two slightly different systems, both utilize the same approach. There's two AAV8 vectors which encode all the components of the system. One of these AAVs encode a cDNA sequence of the lysosomal enzyme of interest. In this presentation, it's beta-galactosidase. And the second AAV vector, which it actually encodes the Cas9 nuclease, and it's under the expression of a liver-specific promoter, meaning that any genomic modification should only occur in the liver. And now looking at the right half of the slide, I'm showing you what occurs if both AAV vectors enter the same hepatocyte of the liver following IV injection. So in hepatocytes, a specifically designed guide RNA, like Dr. Ra introduced, will form a duplex with a Cas9 enzyme and bind to and create a double-stranded break in the DNA in which the cell will want to repair naturally. The idea is that during this repair, the cell will utilize the second AAV sequence that contains the lysosomal enzyme of interest to repair this break, resulting in integration into the genome. And in this case, it's going to be intron 1 of the albumin gene. So albumin is chosen because it is the most abundant protein in your bloodstream, with your liver generating approximately 15 grams a day, or 15 grams of it per day. And if successful, based on the design of the system, the end result is going to be a secreted lysosomal enzyme that can be endocytosed throughout the body and is utilized by deficient cells to break down accumulating macromolecules. Next slide, please. In the last couple of years, our group has applied this technology to murine models of both MPS I and Sandhoff disease, both showing therapeutic benefits. Next slide. But for today's talk, I'm going to focus on how I've been utilizing the PS system to treat the mouse model of GM1 that I created and characterized and published in 2019. Next slide. So for this study, I utilized 2 different lysosomal transgenes, both encoding a different version of beta-galactosidase enzyme to compare their therapeutic outcomes. In one of the groups of animals, I utilized a donor sequence that encoded the human beta-gal sequence, which I'm terming throughout the presentation as HS-GLB1, which is short for Homo sapien GLB1. In the second group, I utilized a donor sequence that encoded the mouse beta-gal sequence, which is termed here and throughout the talk as MM-GLB1 for mus musculus, which is just mouse GLB1. And so in both groups, the second AAV is going to be encoding the cDNA of Cas9 and the guide RNA that's targeting intron 1 of albumin. Next slide, please. So for this study, neonatal GM1 mice were intravenously treated with either human or mouse beta-gal PS system between day 0 and day 2, meaning they were injected before 2 days of age. So mice received 1 of 3 doses, which are going to be termed low, mid, and high throughout the presentation. And because the onset of disease of these mice is usually between 4 and 6 months of age, the animals were allowed to age. And during that aging process, we collected blood and measured beta-gal enzyme activity in the plasma every month until we conducted behavior tests at 6 months of age, which included the beam walk, accelerating rotor rod, the pole test, the Barnes maze, and inverted screen. And so between 8 and 10 months of age, when the GM1 mice start displaying a more significant phenotype, the animals were euthanized and the tissues are collected with various biochemical and histological analyses. Next slide, please. So the first data I'm going to show you here is the beta-gal enzyme activity in the plasma over a 7-month period following treatment. And so of note in the presentation, the red hue colors are going to indicate animals that received the human beta-gal, and the blue hues are going to be indicating the mouse beta-gal. If you're colorblind, I've also used or have trouble with colors, there's different shapes for each one. And so here you can see that there's this clear dose-dependent response of enzyme activity where a mouse that received a higher dose typically had higher levels of enzyme activity compared to those that received the same sequence at a lower dose. But more importantly, what we see is that even at the same dose, the mice that received the mouse beta-gal sequence had a significantly higher enzyme activity than mice that received the human sequence. And this is especially evident when you look at the high dose, where you see that there's almost a 10-fold difference in enzyme activity between the mouse and the human beta-gal sequences. Next slide, please. And so in the tissues, we also observe this dose-dependent response. Since this therapy is liver-directed, it's no surprise that there's a high level of enzyme activity in the liver. And in the heart and the spleen, we also see a significant increase in the beta-gal enzyme activity in the groups that receive the high dose of beta-gal. In all tissues, though, there's this clear evidence that the mice receiving the mouse beta-gal sequence had higher enzyme activity than the animals that received the same dose of the human sequence. But more importantly, we also observed a significant increase in beta-gal enzyme activity in this top right corner in the brain of the mice that received the high dose of mouse sequence. Next slide, please. But with the high enzyme activity, we wondered whether these results, or this would result in a decrease in pathogenic substrates such as GM1 and GA1 ganglioside in the tissues. And while it's not significant in all the cases, we see that the levels of ganglioside in the liver, heart, and spleen are reduced in most samples that were analyzed. Next slide, please. But the tissues but those tissues have low levels of gangliosides at baseline naturally compared to the brain tissue, which is abundant with gangliosides. And so when this project first started and I was using the human sequence, the brain ganglioside analysis was conducted just on a mid-sagittal section, so a section straight through the center of the brain. And however, when we analyzed this tissue, we didn't see any conclusive information or decrease in ganglioside content. And so when the mice that received the mouse beta-gal sequence were euthanized, we decided to microdissect the brain and analyze samples from relevant areas to the behavior tests that I conducted, including the cerebellum, the hippocampus, and the cortex. The analysis showed that the observation of a slight decrease in GM1 content in the cerebellum and GA1 in the hippocampus of the animals that received the high dose of mouse beta-gal sequence. Next slide, but due to, I'm going to skip the next slide just for time. Thank you. So as I said at the beginning of my talk, the animals are subjected to many behavior tests starting at 6 months of age. In the first slide, I'm showing you the results of the beam walking test, which analyzes the mouse's motor function and coordination. So over the course of 4 days, the animals are trained to cross a beam and improve on their time and improve on the number of foot slips on their hind limbs over those days. Normal mice try and escape this bright light that's at the beginning of the test and cross the beam and enter a dark enclosure on the other side, where a GM1 mouse typically has very high difficulty performing this task, including increased time across the beams, as well as the number of foot slips that it has as it crosses. But in treated mice, we see that the animals that received a high dose of the human beta-gal sequence performed better on day 2, but on subsequent test days started performing more similar to a diseased animal in the time that it took to cross the beam. However, the data shows that the number of foot slips that these animals had did improve each day, showing some improvement. When we look at the mice that received the mouse beta-gal sequence, we see that they had a significant improvement in their ability to cross the beam in both the time to cross and the number of foot slips. When we subjected the animals to the beams of increasing difficulty, which we see on the bottom here, we start pushing the limits of their motor function. And you can see in most cases, and most instances actually, the mice receiving the high dose of the mouse beta-gal sequence cross the beam in a similar time to a normal mouse. When we look at the number of foot slips in these animals, we also see that the mice receiving the high dose of both had improved motor function. Next slide. Actually, do 3 more, sorry, 1, 2, 3, yeah. There you go. So I'm going to skip to the bottom right-hand corner because the 2 tests on the left-hand side and the top right corner are looking more at motor function where you see improvement. But the last test that we did was looking at cognition, which is the Barnes maze, and measuring how long it takes the animal to learn to find a hole and escape this platform that they're scared to be on. And we see that animals that received the high dose of the human sequence performed really well on day 3, but on day 4, they did revert back to a more cognitive decline phenotype. They forgot how to complete the task. Whereas a mouse that received the mouse sequence actually performed very similar to a normal mouse, showing an improvement in cognition. And so next slide, please. So just quickly in summary, while running out of time here, is that even at the same dose, the mouse sequence provided higher levels of plasma enzyme activity than compared to a human sequence, including in the brain of the mice that received the mouse sequence. Both of these approaches prevented the onset of motor and cognitive decline in beta-gal mice, as typically observed at 6 months of age, but to varying degrees. Overall, it seems that the mouse beta-gal sequence provides a more beneficial therapeutic outcome in GM1 mice and may be relevant for future therapeutic development. But this data also supports that the PS gene editing system can serve as a platform technology to treat various lysosomal disorders. Next slide, quick, just as an acknowledgement. So here's my acknowledgements. I want to thank everybody that helped with this, and thank you to everybody watching. If you have any questions or comments that don't get answered in the comment section, please take a picture of this QR code on the bottom right-hand corner, and I'm always open to conversing about these projects, any other projects, or any questions you might have about this field. Thank you for your time.
Thanks very much, Michael. And as a reminder for everyone participating, if you have any questions for any of the speakers, we will have time at the end for Q&A. And so now I will turn it over to Michelle Miller from the University of Pennsylvania Orphan Disease Center to talk about their natural history program. Thank you.
Hi, thanks everyone. The title of my talk today is Your Children and Our Quest for a Cure. Next slide, please. So before I go into the actual natural history study, I think what I've heard from the community, not only in this setting in previous years, but more broadly is, oh, another natural history study. So I feel like as a scientific community, we're not explaining very well to the broader community why these studies are so important. So I wanted to take a moment to highlight the importance of these studies. And I'll start with a quote from my favorite scientist, Albert Einstein, which says, to raise new questions, new possibilities, to regard old problems from a new angle requires creative imagination and marks a real advance in science. So our goal is always to advance science, and to advance, we oftentimes have to keep looking back at the old information and saying, what can't we understand? How do we figure out? What new questions do we ask? So in practical terms, what does that mean? And I've listed a number of practical applications in the slide, in the lower half of the slide. So this means we take that information to say, what assessments can we create that we can use in an interventional trial that will tell us if a drug is working? When do we do those assessments? When is the best time to look at changes in biological markers, changes in imaging, changes in motor development? Looking at biomarkers themselves, a lot of people use the term biomarker very loosely, but a true biomarker is not just something that changes in your blood as a result of a disease, but it's something that changes and is predictive of the clinical response. So it's linked to the clinical response. So we're constantly looking for those types of biomarkers because typically they change long before the physical or visual component of change appears. So it lets us know sooner that something is working. We look at current treatment modalities and how they may affect or influence long-term progression or the current status of disease among various patients in various regions of the world. And skipping to the last point, and what's most important for my trial at the University of Pennsylvania is these data are actually serving as a reference arm in an ongoing interventional trial. So the data from the patients who've received an investigational product will be directly compared to what we see in natural history. And that is important for regulators to understand whether or not the product is working. So next slide. So to just go over a little bit about what's going on in the natural history study since I last spoke about this. So just to remind folks, this study is expanding disease-specific knowledge related to demographics, different types of mutations that different patients possess, and whether or not those mutations influence rates or patterns of disease progression, which are measured through a variety of physical and biochemical assessments that are listed there. We look at feeding complications, ventilation requirements, and again, as I mentioned in the previous slide, there's constant biofluid collection to evaluate novel biomarkers. And just to give you a real life example of, again, how natural history helps influence the progression to a treatment and hopefully a cure. Since this study started, we have already eliminated 2 biomarkers that we originally began collecting because we have since learned they don't provide valuable clinical information with regard to disease progression. So in the orange box there, just to summarize, we're trying to find information that will help us predict progression and outcome of disease reliably. When we measure those things and how we measure them, can we find things that will help us predict a change early? And when we find those things, are they actually things that we can put into place in a clinical interventional trial? Next slide. So for the GM1 natural history study at Penn, pretty much to get into the study, you have to have documentation of the beta-gal enzyme activity reduction. We do collect GLB1 mutations, but if you don't have those data, we will run the assay required to collect those data at no charge to study subjects. An important point because I think it's been a misunderstood point for a long time. Under exclusion criteria, if your child is currently enrolled in a trial using an investigational product, they cannot come into the natural history study. However, if a child is not currently enrolled in a study with an investigational product, they can come into the natural history study. And if at any point during their participation in the natural history study, they want to join a trial using a treatment, they can leave the natural history study. Participating in the natural history study does not preclude the child from going elsewhere. And of course, any condition, other exclusions would be any condition that could be, put your child at undue risk. Next slide. So in our study, we have study centers globally. In the United States, we have CHOP in Philadelphia, UCSF Binoffs in California. Canada, we have McGill, Brazil, Porto Alegre. In Turkey, we have Gazi University and Great Ormond Street in the UK. Last year, somebody did ask me to go to Australia. We did meet with that investigator several times, and the COVID restrictions over the last few years with that institution essentially has made it impossible to bring them on board at this time, but hopefully in the future. Next slide. So as of earlier this month in our study, we have 23 patients enrolled globally. 11 have the juvenile form of GM1 and 12 the infantile form. And again, going back to that criteria about participating in an interventional trial, you will see in that third bullet down that 2 of those 23 patients have withdrawn from the natural history study to participate in an interventional trial. 6 patients, unfortunately, are deceased. We're still enrolling. Our enrollment goal is at least 40 patients by June of next year. So we're open. Come on in, join us. We'd love to have you included. Next slide. So at a high level, our goals are to build a robust data set, not only for us. And I think one of the unique aspects of our trial is that we have made a commitment to share our data with any researcher, foundation, or commercial entity that makes a valid scientific request for the data. Next slide. And in closing, I would just like to thank first and foremost our amazing and heroic patients and their families, without whom we would have none of these data. And of course, the physicians serving as PIs for the trial. And again, Christine and all of the Cure GM1 Foundation for continuing to support us. Thank you.
Thank you so much, Michelle. We'll now turn it over to Dr. Georgino from Azafaros.
Thank you very much. I think I need to start the video if I'm allowed to, but I can start. So the topic, and thank you very much to Christine for giving Azafaros the opportunity to be here today, especially in this session after eminent speaker from the academy. Christine, I think that all the community needs to thank you for all the great efforts that you're doing. Also, the meeting that you are planning for Friday, I think is extremely important. So I don't know if you can see my face. In any case, you will not lose anything. But Dr. Miller before, if you go to the next slide, has already anticipated the need not to give up, in a way that there is certainly the need to gain more data, more information. When we talk about natural history study, we include a large set of potential design. Of course, one could be to collect data which are already there in the medical chart. Another one that we always see in single-site studies is that we have scattered information over time. There is not really a unique assessment, a schedule, or a timing of the visit. So what we are trying to do is to design a study that is feasible, where the there is enough convenience for patients, but at the same time also where we want to harmonize in a single protocol a series of intervention that would allow to collect in a consistent way prospective data. If we go to the next, thank you. So the acronym of the study is PRONTO, and in the study there is no experimental treatment. In a way, is an investigational and is an interventional study, because we are asking all sites to try to conduct the same type of assessment, and I'm going to spend a second. The SARA scale is a little bit the core of the assessment in this study. The inclusion criteria are we are fundamentally including patients who have a late infantile or juvenile onset of GM1 gangliosidosis or juvenile onset of GM2 gangliosidosis. So also because we would like to explore further the similarities in some cases of clinical phenotype of the two conditions for potential future studies. If you go to the next, thank you. This is a little bit the status. The study has started this year. We have selected 13 sites in North America, in the US, Brazil, and in Western Europe. 12 of the 13 sites are activated, and so far we have enrolled 12 patients. If you can go to the next one, please. So the inclusion criteria are the one that you see in the slide. So we can enroll both GM1 gangliosidosis and GM2 gangliosidosis, i.e. Tay-Sachs and Sandoff, chronological age 2 to 20 years, with the definition of disease onset, so that they had a normal development until the first birthday, and then they started having neurological manifestation after that, and who has a patient who has at least abnormal gait and speech of any severity at the beginning of the study. If you go to the next, please. The assessment, so this slide is the graphic layout, it's not ideal. We have a clinical assessment, and the basis is the SARA scale, so the scale for assessment and rating of ataxia, which can be done also remotely. So there is a telemedicine platform which has been put in place. The INAS, which is conducted at the site, where the investigator is also looking at other neurological signs different from other than the ataxia one. The MFM32, which is a validated scale for looking at motor function measure, the time up and go test, so the time that the subject need to stand up, walk for 3 meters, go back and sit again. So these are mostly clinical assessment, but we have also put in place some electronic diaries. For instance, we have different language version of the Vineland Adaptive Behavioral Scale, and also the Barden scale for a family caregiver. So this is going to be important also to characterize some aspect related to quality of life. And finally, we have also digital endpoints. So a digital sensor is a patch that can be worn on the trunk of the child, and through a mobile phone, it's sending data to a central server where we're going to apply, thanks to a partner, which is a specialized AI company, to evaluate different aspects of gait and balance. If you go to the next, please. Yeah, I think something very great that we have done, also with the partnership of several patient associations, including GM1, but also CATS and Actais in the UK and in Spain, and the NTSAD, is to develop a video. It has been really a great enterprise where we are trying to explain to patients how the PRONTO study is designed, and the assessment that the study foresees, I hope that you had or you will have the time to have a look. I think it's very is a very interesting way of for easy communication, also to those of us who are not familiar with the clinical research setting. So now, if you can go to the next slide, I would like to give also, because this is the natural history, a little bit some information on our lead project, which for the time being has only a code, which is AZ3102, and to talk a little bit about Azafaros. If you can go to the next slide, please. So Azafaros is a startup company. We are a limited number of people, and it was founded in 2018 in Leiden, based on a series of patents of molecules coming from the University of Leiden, and in particular the chemistry and biochemistry department of where several experts in the field are working since a while, including both Hans Ars and Hermann Hovercliff. So they were looking since a while at molecules, and in particular aminosugars, that could selectively and potent inhibit two enzymes which are involved in the metabolism of glycosphingolipids, and in particular of glucosylceramide. One is GCS, so glucosylceramide synthase, which is an enzyme that many of you know because it's in the concept of the substrate reduction therapy. But another enzyme which is also important is the neutral non-lysosomal glucosylceramidase. I think we will have time in the future to explain, and we are also trying to develop a video to explain the mechanism of action. But the idea was to select a molecule which was, as I say, potent and selective. And if we go to the next slide, please. If we try to make a comparison between our molecule and miglustat, we do clearly see, and we show the IC50. Now, for those of you who are not familiar, the lowest the IC50 number is, the more potent the molecule or the drug is. So you see that AZ3102 is extremely potent at inhibiting both GCS and GBA2. So if we compare it with miglustat, for instance, it is 10,000 more potent at inhibiting GCS, and I would say between 250 and 500 times more potent at inhibiting GBA2. But on the other side, what is also important is the lack of inhibition of the intestinal disaccharidases, which are those enzymes, especially sucrase and maltase, that miglustat is actually inhibiting. And so all the cascade of events such as diarrhea, due to the accumulation of sugars in the intestine, which are important limitation factors in terms of gastrointestinal tolerability for miglustat. So the molecule, AZ3102, is potent, selective, so it's devoid of potential GI tolerability issue. This is what we have seen in animals and also in healthy subjects who have been receiving the drug so far. If you go to the next one, I can anticipate some information. So last year, I would say beginning of this year, we have completed a phase 1 study in healthy subjects, as usual in the development of this oral small molecule. So we started from a single dose, starting from 1 milligram. We were able to increase the dose pretty quickly because the safety and tolerability was very favorable. And then we administered 9 milligrams. So you see the very low doses for 14 days. The adverse event profile was extremely favorable, benign. So we didn't see diarrhea or GI discomfort. We haven't seen some events such as appetite suppression or weight loss that has been reported with miglustat. No reduction of platelets. There were no effect on the ECG, nor tremor or other neurological manifestation. It's important to emphasize that these are healthy subjects. And the only event we have seen is some skin, I would say epidermal effect of mild intensity, but these were very, very expected based on the mechanism of action. And we are confident this will not be a problem in the future studies. Now, if we go to the next, thank you. A little bit of the pharmacokinetics, so how the body is managing the product in a way. So we have seen a very rapid absorption, and also important because it's very high, it's around 90 to 95%, so very good. There is no accumulation, which is an important information over time, over multiple doses. And we've been able to reach a level of exposure similar to the one that we have been able to reach in an animal model of Sandoff disease, where we saw a benefit in terms of survival. Now, of course, this must be confirmed in clinical trials in patients, but this is quite reassuring. And importantly, it's the kinetics would allow a once-a-day administration, so this would be also a factor to improve adherence and compliance to treatment. And what we have seen also is a clear pharmacodynamic effect. So what you see in the slide are changes of blood level of glucosylceramide, but we have seen a similar pattern for other glycosphingolipids. And as you may notice already, after 4 days, there is a separation of the two lines. So the median reduction at the lowest level is around 70%. And what you see also at the end of the curve is that as soon as the drug is discontinued, so after the last dose, the level of glucosylceramide are catching up, are going back to pre-treatment level. So this is also important in terms of potential safety. Now, to go to the next slide, and which is my last slide, and thank you very much for the patience that's listening, is a little bit what we are planning for the next months. So the important thing for us is to keep recruiting patients in the pronto. That's extremely important. We acknowledge the need to collect more and more information. We're going to start testing the product in patients, in a small group of patients, to start getting information on the safety in patients and the pharmacokinetics. This would also allow to select the right dose for the pivotal study. We still need to completely characterize the metabolism of the drug, and this is something that we are doing at the moment. We have started discussion with the health authorities, in particular, as always, with the FDA. And so 2023 will be also dedicated to design altogether, because we want to have also the patient community involved in the design of the study, and to start the study by the end of this year, or of next year, I'm sorry. So that's my last slide. Thank you very much.
Thank you very much, Ruben. Much appreciated. Just another quick reminder to everyone that we're going to have some time at the end of everybody's talks for Q&A. So you can put them in the chat, or you can wait and raise your hand during Q&A. And now I think, Jeff, we have a video presentation from Dr. Tratt at Lysogene. Great.
Good afternoon. I'm Dr. Marie Tratt, Chief Medical Officer at Lysogene. I'm here today with you to present our program and therapy list GM101, which is an innovative gene therapy targeting the treatment of GM1 gangliosidosis. GM1 gangliosidosis, is a fatal autosomal recessive disease caused by mutations in the GLB1 gene encoding for the lysosomal beta-gal enzyme. The deficiency of that enzyme leads to the accumulation of GM1 gangliosides in neurons with progressive neurodegeneration. Currently, there is no disease-modifying therapy available for the treatment of GM1 gangliosidosis, and any effective treatment would be expected to achieve delivery of active beta-gal protein to the CNS, both brain and the spinal cord, where the major pathology is observed. Lysogene's investigational medicinal product is an adeno-associated viral vector serotype RH10 carrying the human GLB1 gene formulated as a suspension for injection into the cisterna magna as presented at the lower bottom right of the slide. The clinical trial is an interventional, multi-center, single-arm, two-stage adaptive design study. In the first part of the study, which is first in human, enrolled subjects with infantile GM1 gangliosidosis will receive a single dose of the therapy by intracisternal injection. Once all subjects have been treated and have reached at least 6 months of follow-up, an interim analysis will be performed to look at all of the safety data and preliminary efficacy data before moving into the next stage of the study called stage 2, which will focus on collecting efficacy data. In that second part of the study, around approximately 12 additional patients with infantile GM1 gangliosidosis will be recruited and treated. And at a later stage, it is as well planned to add patients with juvenile forms of the disease. In stage 1, the study that has now completed recruitment, the main endpoints were focusing on safety. So incidences of treatment emergent adverse events and treatment emergent serious adverse events were collected, as well as any changes in general physical and neurological status, changes in biological parameters and vital signs, assessment of humoral immune responses, as well as data from evoked potentials to monitor any sensory deficits. Efficacy data were as well collected, and the two main efficacy outcome measures were percentages of patients achieving or maintaining major motor milestones such as head control, ability to sit, crawl, or walk and stand, as well as overall survival and event-free survival will be collected, time to lose previously acquired motor milestones, as well as time to gaining the next major milestone, as well as magnetic resonance, MRI, volumetric measures. In the first part of the trial, 5 subjects have been recruited and treated, of which 3 early and 2 late infantile patients aged 5.5 months to 24 months. The safety and tolerability data reported in this presentation cover a period spanning from May 2021, when the first subject had been enrolled and treated, to date, which is October 2022. All medical and safety data were reviewed on an ongoing basis, including medical history, concomitant medication, physical and neurological statuses, collection of adverse events, and any related INP-related events were scrutinized and reviewed closely. The gene therapy was overall very well tolerated in the perioperative and postoperative period. No treatment-related serious adverse events have been reported to date. There were 5 SAEs, all reported by the investigators, considered as non-treatment related and more likely related to the underlying disease or an intercurrent condition. The 5 SAEs were leg cramps, 1 case, 2 viral infection cases, those were gastroenteritis and pneumonia, 1 case of seizure leading to hospitalization, a stridor with respiratory insufficiency, were all, as I said earlier, considered as non-INP related by neither the principal investigators nor Lysogene. A Data Safety Monitoring Board will meet by the end of this month to review cumulative data of all patients up to the minimum of 3 months post treatment for the last patients enrolled. An interim analysis, I said earlier, will also occur once the last patient has achieved the 6-month milestone, and this interim analysis will occur in Q1 2023 to look at all the data, as I said, that will inform the next stage of the study. In conclusion, the ongoing safety data review of the 5 subjects enrolled in the safety cohort, the safety first in human part of the trial, demonstrates that this GM101 gene therapy and the procedure linked to injecting the product into the cisterna magna was overall well tolerated with no incidence of any treatment or procedure related events. All SAEs reported during the observational period do not relate to the INP as assessed by the PIs and lysogene. The interim analysis in Q1 2023 will allow for the analysis mainly of the safety data and the preliminary efficacy data that will inform and will allow moving to the next stage of the disease, which is of the trial, sorry, that will occur in H2 2023. I would like to acknowledge all the principal investigator with a big thanks to the patients and their families that have participated in the trial, as well as our colleagues at Cure GM1 and the National Tay-Sachs and the Lyme Disease Association, as well as my colleagues at Lysogene. Thank you very much for your attention.
Great. Thanks for sharing that video, Jeff. So now we'll turn it over to Dr. Weinstein of Passage Bio.
So, good afternoon, everybody. Can you see me? Because I can't see the screen. Are you able to hear me and see me?
You're good.
Okay, perfect. So good afternoon, everybody. I'm David Weinstein. I'm the medical lead for the GM1 study, which is the trial of GM1 to treat GM1 gangliosidosis. For those of you who don't know me, I came to Passage Bio after 30 years in the academic world. I came to Passage Bio because I had the privilege of living through what Dr. Gascock talked about earlier today, seeing children with a serious, rare medical condition get better. So I was doing glycogen storage disease work. And when I started, most of the children were getting liver transplants. And with the power of gene therapy, those not only have we been able to treat people, but some of the children and the adults are off treatment completely. And I'm happy to say that over 4 years later, some of the patients are still off treatment and doing great. And it's because of that dream of seeing the children be healthy that I came to Passage Bio to work on GM1. Now, you'll recall when I spoke 2 years ago, it was still a dream. We did not have approval from the FDA. We were just presenting ideas for our trial, and we were trying to get approval to do it. Last year, when the trial was presented, we presented that we had dosed our first patient. And the reason I'm so excited right now is I get to report that we are now almost done with our dose escalation trial. We not only have we treated 1 patient, we've now treated 7 patients, and I'll go through that with you. But we are on target for finishing the dose escalation, the first part of the trial, by the end of this year. And that makes me so excited because the trial is moving along very efficiently. Now, next slide. I would like to just emphasize that this is not an approved trial, that we are still in the phase part of the study looking at safety and efficacy, and we're trying to learn how to do the gene therapy, how to do it safely, and how to maximize the benefit of it. This is a totally researched trial. The safety has not been established in humans, and it has not been approved by any of the regulatory agencies. Now, in the past, we have gone through the study design, and in the interest of time, I will not be going through that this time, but I'll refer people to the clinicaltrials.gov listing, which gives you all the studies and all the outcome measures that we will be looking at. Next slide. So for those people who haven't heard me before, haven't heard about our work, we are using a next generation AAVhu68 vector to perform gene therapy, and you've heard a lot about gene therapy. We're using the virus to deliver the GLB1 gene encoding beta-galactosidase to the brain and to the peripheral tissues to treat children with infantile and late infantile GM1. Next slide. We are using the cisterna magna, which you have heard a little bit about. The cisterna magna is the space between the bottom of the brain and the spinal cord. And while that sounds scary to insert a needle and do the injection there, there's actually a space, as you can see in the figure there, which allows the virus to get injected and go throughout the brain, because the brain is the most important organ we're trying to treat. Of course, we also want to treat the other organs, and we believe we can do that, but we can also get the best treatment for the brain. Now, this procedure has been used for over a century, but it can be done much safer now with modern technology, which allows imaging and very precise placement of the needle. Next slide. The reason Passage Bio has chosen to use the cisterna magna injection is we think this is the best way to treat the brain. I presented data 2 years ago showing studies from the animal models, which shows that the cisterna magna injection leads to great delivery throughout the brain and treats the brain better than any of the other techniques that have been studied. The reason it can do this is because it bypasses a barrier called the blood-brain barrier. So that's something that has limited treatments in the past. And so we inject directly into the spinal fluid, and it goes to the brain and can treat it. Another advantage to using the cisterna magna is that we are not impacted, we think, by pre-existing antibodies. So Dr. Riley mentioned that in his talk earlier, that one of the challenges with gene therapy is that the body could have seen the virus and have antibodies against it. And this technique is not impacted because the antibodies don't cross into the CSF. We also think by giving it this way, we'll have less risk of liver toxicity, which again was discussed earlier as one of the limitations of gene therapy. And we think that this can be done very safely because of use of radiology techniques. And in fact, data using the ICM procedure was presented at the World Conference earlier this year, showing 27 children successfully getting gene therapy using this technique without complications. Now, there are some challenges. The first thing is because of the technique, we have to use anesthesia, and we don't like to expose children to any risk or any procedure unless it's necessary. This does require additional training because most doctors haven't done it, and there is a risk of bleeding, but we feel that those challenges are minimal compared to the advantage of getting great gene therapy to the brain. Next slide, please. So let me give you an update of where we are and an overview of our trial. So this is a phase 1/2 multi-center open label dose escalation and confirmatory study. So what that means is that we are trying to learn about this drug to find out, does it work and is it safe? And safety and tolerability were the principal goals of the first phase, finding the right dose for the treatment and also finding out who benefits from it. One thing to note about this approach that we're using is that we get to use very low immunosuppression. So oftentimes when gene therapy, other techniques have been used in the past, it requires lots of drugs to prevent an immune response, but because of the injection directly into the spinal fluid, we get away with very low dose immunosuppression, and we're only doing low dose steroids for 4 weeks, and then we taper off. So the goals are safety and tolerability, and of course, we're trying to learn and get efficacy results. When we presented to the FDA, they wanted us to get more experience with this, so they asked us to start with the late infantile population. So we dosed our first patient with our treatment in March of 2021, and that was a late infantile patient with the low dose. After treating 2 patients, we went to the safety committee. They had no concerns, and we were able to go to the high dose for the late infantile, and we went to the low dose for the early infantile. We treated our first patient with early infantile in January of this year. And as you can see depicted here, we have now treated everybody in cohorts 1, 2, and 3. One thing to note, which we talked about also 2 years ago, is that if there are safety concerns, we will expand the cohorts. And so you can see we have used the minimum number of patients because it has been well tolerated. We are now recruiting in the high dose early infantile cohort, cohort 4, and as aforementioned, we are on target for finishing this and finishing the safety and efficacy, this part of the study, by the end of this year. Now, I would like to make a comment here about the next phase. So once we finish this, figuring out the dosing and showing the safety data, we have to go back to the FDA. The expansion cohort is ultimately the trial, which will lead to approval or not getting approval. And that's where we have to prove that this drug is efficacious. Now, we have to go back to the FDA to get permission and to review what we're going to do for that, for that part of the study. And there will be a gap, and this happens in every study. And so there will be a period from the time we finish dosing our patients in cohort 4 until we can go into the expansion cohort while we get permission from the FDA. Now, I'm a pediatrician, and everybody at Passage Bio cares about the patient. So you have my pledge, we're going to go as fast as we can to minimize the delay so that we can get back to treating patients as soon as possible. And we expect that will happen in 2023. Next slide. Now, I present here data from the 7 patients that have been treated. One of the goals of an early trial like this is to learn about your drug. So we purposely have tried to collect patients of various severity. We have early infantile, we have late infantile, we have people who are early in the course, like patient 1 who was relatively early in the course, and we have patients who were not diagnosed till very late. So you can see the difference between patient 1 and patient 2. One was diagnosed, they were diagnosed at a similar time, but actually I see a typo, but they were, the age of gene transfer was much later because the child, patient 2, was not diagnosed until 30 months of age. Another thing I'd like to point out on this slide is that we have a range of severity. So, patients 3 and 4 were very severe. So if you look at the developmental age, you'll see that patient 3 was very debilitated, had regressed, and was down to 2 weeks developmentally. And we want to learn about what does our treatment do in both the severe and the less severe patients. And that's why we have included people across the spectrum in our study. In contrast, patients 6 and 7 were people who were less severely impacted. And so we're going to learn a lot through studying this population. Another thing I'd like to point out is patient 1 had antibodies. So in most studies that are done with, for instance, intravenous gene therapy, if someone has pre-existing antibodies, they can't get the treatment, but patient 1 did have pre-existing antibodies and still was allowed to participate. Next slide. So just to summarize, because 10 minutes is not enough really to go into the data, and we also wanted to make sure that we get the latest results from the study, the patients being studied right now. We are on target for completing the dose ascending phase of the study by the end of this year. We've had a very positive safety profile. The treatment's been very well tolerated. There have been no treatment-related SAEs, severe adverse events, and we've had no complications related to the ICM injection. Now, I'm very, very excited to share with you the developmental biomarker radiology studies from these patients, and we plan to release more data by the end of this year as soon as we have the most recent data, and we will also plan to submit it and present it at the World Symposium, which will be occurring in February 2023. So let me end, next slide, by again doing a plea for the natural history studies. None of us want to do a placebo-controlled trial. In order to do this, we need to, in order to convince the FDA, we have to have good natural history data to compare. And you've heard this over and over again from many different people. So please, consider going into the natural history studies. And also, again, you've seen from Michelle's talk, participation in natural history studies does not limit participation in the treatment trials. Patients come first, and of course, we would never restrict someone from going into a treatment trial to participate in the natural history study. Finally, next slide, I just want to say thank you to the fantastic investigation team. To me, this is the equivalent of the dream team. If anybody's a basketball fan from the Olympics, we have the great investigators from around the world, and we've had patients participate from around the world. I also want to acknowledge the University of Pennsylvania, who did all the preclinical work for these studies. I am an employee of Passage Bio, so I will acknowledge Passage Bio support. And finally, next slide, most importantly, we want to say thank you to the families, to the Cure GM1 Foundation. You have inspired us. We appreciate your trust, and thank you for allowing us to do this work.
Thank you, Dr. Weinstein. We have one more talk, and switching gears now to talk about enzyme replacement therapy for GM1. We have Dr. Jillian Brown from Tega Therapeutics. Thanks.
Thanks, Dawn. Hi, everyone. Like Dawn says, I'm Jillian Brown. I'm the Director of Research and Development here at Tega Therapeutics. I would like to thank you for the opportunity to talk to you today about our collaboration with the Cure GM1 Foundation. Together, we're working to develop an enzyme replacement therapy as a potential treatment for GM1 gangliosidosis. We are working with the GM1 knockout mouse that was created by the Jackson Laboratory, and today I'll provide an overview, a very short one, and the progress of some of our work here at Tega. Next slide, please. GM1 gangliosidosis, as everyone knows, it's the lysosomal storage disease that is caused by the loss of the lysosomal enzyme beta-galactosidase. And as a result, toxic accumulation of the complex sugars known as GM1 gangliosides occurs in the brain and the systemic tissues. And it is this severe accumulation that causes the neurodegeneration, the seizures, and the rapid developmental regression. And as been mentioned already in the talks today, there is currently no treatment available. Intravenous enzyme replacement therapy resolves somatic issues. However, as we've also talked today, complications still exist in the brain because the enzyme is unable to cross the blood-brain barrier. Next slide, please. The barrier prevents the uptake of these large recombinant enzymes to the brain. And as we've talked again, the symptoms of the central nervous system and lysosomal storage diseases in general are therefore very difficult to treat. The good news is intracerebral ventricular is a delivery approach that is safe and effective for delivering enzyme directly to the brain. We could go to the next slide, please. And this does so by bypassing the blood-brain barrier. It delivers the enzyme directly into the cerebral ventricles, as shown here. It uses a surgical device that's implanted underneath the scalp. Actually, chemotherapeutic drugs are routinely delivered using this device, and so it's considered safe and very effective. Brunera is a lysosomal enzyme, and this was the first approved safe and effective intracerebral enzyme replacement therapy. It is used for treating CLN2 Batten's disease. And there is also an ongoing phase 2 clinical trial in MPS IIIB patients that delivers a modified lysosomal enzyme, and it does this intracerebroventricularly also. So we here at TEGA, together with the Cure Foundation, think that this enzyme delivery approach should be safe and effective for treating children with GM1 gangliosidosis. Next slide, please. The human beta-galactosidase, again, has been mentioned today, and it's the enzyme that removes the beta-linked lactose on the end of the complex glycans, as shown down in the right-hand corner of the slide. The GLB1 gene makes the enzyme that breaks down the GM1 gangliosides inside lysosomes. And the GM1 ganglioside patients are missing or have a faulty GLB1 gene and therefore cannot make active enzyme. This results in toxic levels of the GM1 gangliosides in the brain and the spinal cord, and it's what causes the severe damage. Next slide, please. Clinical disease phenotypes, the severest form being the infantile onset. It has very low levels of the residual beta-galactosidase enzyme activity, and the developmental delay is within the first year of life, with rapid loss of motor skills and death by 2 to 4.5 years. Late infantile, juvenile, and adult onset have higher levels of the residual enzyme and a slightly longer survival rate. The residual beta-galactosidase activity range is very narrow among different patients, and it ranges from 0 to 15% of normal control levels. Therefore, minimal restoration of enzyme activity is needed to reduce the GM1 ganglioside buildup and prevent disease progression. Next slide, please. So the first question that we asked, can we deliver beta-galactosidase to the lysosomes and restore missing enzyme activity? Mannose-6-phosphate receptors, as everyone knows, on the cell target the enzyme to the lysosome. So GM1 patients deficient in the enzyme, as shown here in the first graph, were treated with different concentrations of the beta-galactosidase enzyme. And the enzyme activity shown here shows that the beta-galactosidase was taken up by the cells and the enzyme activity was restored. On the right just confirms by Western blot analysis the uptake of the beta-galactosidase into the lysosomes also. Slide, please. Jackson Laboratory partnered with the Cure GM1 Foundation to generate a mouse model of GM1 gangliosidosis. Using CRISPR-Cas9, generated a mutant of the GLB1 gene, which carried a 7-nucleotide deletion in exon 5, predictive of a null allele. The mice lacking beta-galactosidase showed a reduction in the beta-galactosidase enzyme activity in the brain, and that's shown in the first graph. The blue bar represents the knockout mouse, the green bar represents the hit litter mate, and the wild-type mice in red. So you can see that there's almost no beta-galactosidase enzyme activity in the mouse. Impaired motor coordination with a reduction in latency to fall from an accelerating motor rod is shown on the right-hand side. In red are the wild-type mice, and in blue are the homozygous, the mice that are missing the beta-galactosidase. And as you can see, as the mice age, they're unable to hold on to the accelerating motor rod. GM1 ganglioside accumulation, however, was not measured in the brain of these mice. Next slide, please. So the question that we had at TEGA was, do these galactoside deficient mice accumulate GM1 gangliosides in their brain? We currently have a breeding colony at the Animal Model Core facility here in town at Scripps Research Institute. And in a mouse natural history study, we collected brain tissues and gangliosides were extracted, isolated, and quantitated. We developed a mass spectrometry analytical method in-house to quantify the GM1 gangliosides. And shown here is just a trace of extracted gangliosides. And in red, you see the GM1, and in green, you see a standard that we spike into the mass spectrometer in order for us to quantitate the GM1 in each of the mouse brain samples. Next slide, please. And the good news is there were high levels of GM1 gangliosides in the brain of these mice. Data showed that they accumulated in the brain. It's the first graph. The blue bar is the mice that lack the beta-galactosidase, and the green is the het litter mates. The middle graph confirmed what Jackson had already shown, that there is in fact no beta-galactosidase enzyme activity in the brain of these mice. And the impaired coordination with the reduction to fall, we saw similar data. And as the mice age, shown here is 12 weeks of age, but as the mice age to 24 weeks, the homozygous mice can no longer hold on to the rotor rod, accelerating rotor rod. So this is a model that definitely shows characteristics of a model of GM1 gangliosidosis in patients. Next slide, please. Studies are underway here at Tega to deliver the beta-galactosidase intracerebroventricularly. Efficacy and safety will be assessed in enzyme dosing studies, and dose-dependent biodistribution of the enzyme throughout the brain will be assessed. Effects on biochemical and histological pathology in the brain will be determined, and motor coordination will also be evaluated. We hope that by replacing the missing enzyme directly into the brain, we can reduce the GM1 ganglioside storage and prevent impaired motor coordination and disease progression. There is evidence in the literature by some of our colleagues that support that this delivery approach will be successful in treating GM1 gangliosidosis. And the preclinical studies outlined here are an important step towards advancing this novel treatment for GM1 gangliosidosis. Next slide, please. So in summary, the mouse model of GM1 gangliosidosis has been successfully established. Significant GM1 accumulation occurs in the brain, and there is no detectable beta-galactosidase enzyme activity. This resulted in impaired motor coordination in these mice, and I think we can all agree that this is promising preliminary data for both biochemical and neurobehavioral endpoints. Dr. Riley just mentioned at the very, very beginning the importance of surrogate endpoints. So these endpoints can be considered good biomarkers, and the biomarkers, as we've talked about today in the talks, will allow for early intervention, be used in newborn screening, and can also be used to monitor treatment in the patients. And as I said, the studies are underway to determine the effect of adding beta-galactosidase intracerebroventricular into the mouse model. I'd like to acknowledge the Cure GM1 Foundation, the Jackson Laboratory, and the team at Tega, and thank you very much for your attention.
Thank you, Gillian. So we have some questions that have been raised and already addressed in the Q&A. Then we have a couple more that are open and a couple that came in through chat, which I would like to share with the broader audience. Maybe, and a lot of them, Dr. Weinstein, be prepared, they're for you. So let me maybe first start with the one that came through chat to Dr. Ra. And someone had asked if you could use CRISPR as a complement to gene replacement therapy. And I thought the broader audience might benefit from that question and answer if Dr. Ra is there. Okay, well, I'm going to go ahead and read the response because I think that people would appreciate it. And while I look for that, Dr. Weinstein, I'm going to start with a question that came to you through the chat that, again, others might not have seen is, would the company consider, would the study consider redosing a patient either within or outside, in or outside the predefined study period if the drug proves effective, but the patient would benefit from a higher dose?
So first of all, Dawn, thank you for moderating, and you're doing a great job. I think that's a wonderful question. Now, we want to do what's best for patients, and we don't know yet. We need more data to determine if the high dose is better than the low dose. And so it is something that, it's possible that we might need a higher dose, and went into the initial patient. So we are very sensitive to that. Redosing is not as simple as it may sound because of the immune response. So the first thing we have to do before we could consider redosing is to get safety data in terms of the immune response. So we talked about how antibodies don't really cross the blood-brain barrier, but once you've done the injection in there, there may be some immune response. So we are in the process of gathering immune data to look at that. And then once we have assessed all that and we see if there's any response, then we can do it. But we have to really continue with the study right now. And then it would mean going back to the regulatory agencies, and it would have to be something totally different. So I want to be careful not to rule it out, but I also can't promise that we're going to do that because we need more information.
Thanks, Dr. Weinstein. Dr. Ra, I was interested in sharing with the broader audience a question that came in via the chat to the hosts and panelists, the question about if CRISPR could be used as a complement to gene replacement. And I was wondering if you could share your thoughts on that. You're on mute.
Some of the more personalized CRISPR strategies probably couldn't be used at the same time or after gene therapy. And that's because, not because of the delivery or anything like that, but because those strategies are targeting those specific areas of the gene. And while they probably wouldn't cause any harm to the transgene, which is being expressed by episomally by the AAV, they just wouldn't be compatible. And so systems like Dr. Prisbilla's, he could probably use that system at the same time as gene therapy, but a more personalized approach probably wouldn't be possible.
Thank you. And we had one other question related to CRISPR treatment. Dr. Ra, are there any known constraints on how old a patient can be when receiving CRISPR treatment?
Yeah, so at this point, the CRISPR that has been used in clinical trials has been solely used for adults. And I think it's just part of that safety and efficacy aspect of, FDA approval and clinical trial processes that are necessary before we can move therapies into children. And so as far as, personalized CRISPR therapeutics, there shouldn't be an age constraint. But we also have to acknowledge that, disease progression can't always be reversed. And for the most part, even CRISPR is only going to essentially stop the disease where it's at. You might see a little bit of restoration of skills lost or some pathology that had, developed neurologically for patients with GM1. But for the most part, we're looking at, stopping the disease where it is and monitoring, developmental progress after that. So, but also just to add for the using CRISPR with other therapies, it would be a great complement to enzyme replacement therapy and could potentially, depending on how efficacious it is in treating a specific mutation, it could replace enzyme replacement altogether or decrease the number of enzyme replacement infusions the patient requires over time.
Great, thanks. And speaking of enzyme replacement, question for you, Jillian, what are the GM1 biomarkers that you are using or that will be used, anticipated to be used in the ERT studies?
Yeah, that's a good question. Right now, we're just looking at the accumulation of GM1, and there's GA1, and there's also other glycans, disease-specific glycans that we could look at. We require a little bit more work to look for standards so that we can use them in the mass spec to quantify them properly.
Great, thank you. So Dr. Weinstein, back to you, a great question. Since ICM administration is not impacted by pre-existing antibodies, does that allow for dosing more than once?
So again, this is sort of a follow-up to the last question. These are great questions. And there's just hasn't been enough experience to know what immune response will occur as a result of this, and if redosing occurs and what immunosuppression would be needed. So it gives hope that potentially down the line, but there's more work that just has to be done before this can be done, because we're just gathering information on the immune response.
Thank you. And a question, I think specifically related to the ongoing clinical trials in gene therapy. I think this was directed also to you, Dr. Weinstein. Does there have to be 60 days between each dose in the expansion category?
So ultimately, this is the decision of the regulatory agencies. So when we have our meeting on Friday, you guys are going to help all of us on this, all of the studies and all of the work, because we need to emphasize how important it is that we get children, and 60 days is an eternity. As was alluded to, we had a 60-day window that we had to go at least 60 days between patients in the dose escalation phase, because for safety reasons. In other trials, the FDA and other regulatory agencies have decreased that, and we're hoping to get the same response from the regulatory agencies due to the lack of safety issues. But ultimately, it will be the decision of the FDA. But we hope that we will not have that 60-day mandated break between patients when we get to the final phase.
Okay. And then another question about ERT, which may be addressed by Jillian and or others, physicians on the call. The question is, can ERT be used for in utero treatment to give the children more time for therapies beyond ERT? I think the main question is, could you conceive of a clinical scenario wherein a patient is treated in utero with ERT?
Dr. Przybyla or Michael chiming in on this one because of just my interest in uterotherapy. There is a possibility for this. There's a clinical trial going on right now by Dr. Tippy McKenzie out of the University of California, San Francisco, that's using ERT in utero for patients with MPS, mucopolysaccharidosis, another lysosomal disorder, but it's multiple different types of mucopolysaccharidoses that she's testing. And that's an active clinical trial that's ongoing right now by administering different doses of specific ERT to that disease in utero before birth and measuring outcomes.
Great, thank you.
There has also been successful use of ERT for Pompe disease, another LSD in utero. And what's been really wonderful about that is for Pompe disease and I'm sure other LSDs, the modulating the immune response for ERT after birth and when enzyme replacement is started is really challenging, especially when enzyme replacement needs to go for the length of the child's life. And so if delivered in utero, the immune response is actually a lot easier to modulate because you're not developing those antibodies that you typically would when you are administered the first dose of enzyme replacement after birth. So.
Great, thank you. And I don't see any other questions, but there is a comment that I certainly want to share as to close out this session. This is not a question, but as someone who took part in the trial, I just want to say thank you. Your work has changed our lives. So with that,
Dawn, I think there was one question, maybe for Dr. Giorgino about the inclusion criteria. Someone said their child is 14 years old with juvenile GM1. I guess they're wondering, like, if you have any information to share about the cutoff for the Azafaros trial.
Thank you, Christine. This is something that we are thinking at the moment. So, maybe in a year from now, we will be able to address this question, and thank you for that more accurately. Yeah.
Okay, thank
But
you.
certainly we are, we are targeting, children with a wide span of age for the future.
Okay, thank you.
Thank you so much.
Great. It looks like that wraps up this session. So we will take
Come
a
back
short
in
break.
half an hour.
Yeah, come back in about 30 minutes. Great. 2 o'clock West Coast, 5 o'clock. Sorry, 1 o'clock West Coast, 4 o'clock East Coast time. Thanks, everybody.
Thank you very much.
Thank you.
All right, it is 2 o'clock. Hopefully everyone is jumping back on to join us for part 3. So part 3 of the conference is the family experiences, advocacy, and palliative care. My name's Kylie Harrison. I am GM1 Foundation board member, and I'm also a mom to 2 little girls that have juvenile GM1. So I'll help moderate this section. And you can see we have another group of wonderful presenters. And so the first half an hour, we're going to be hearing from Emily, David, and Katrina talking about the rare siblings experience and family connections.
Awesome. I think I'm the one starting us out. But hello, everyone. My name is Katrina Erickson, and I am a sibling of Jorgen. And some of you might have met my mom, Cindy. She's pretty active in the GM1 community. But Jorgen has gangliosidosis GM1. He was diagnosed when he was about 10. So I was a baby when that happened. So that's really all I've ever known. I've only ever known my brother with GM1. But I also want to preface my time with saying that I know that every family has a very unique situation with GM1. So I'm just here to share about my experience growing up with my brother, which may not be the same for your children and your siblings. But yeah, I would say with any experience, there's positives and negatives. I remember from a very young age, just knowing that my family was different from other families that I would meet at school. But I didn't always see that as a negative. I just knew that I loved my brother. I loved to play with him. We would play games just like any other sibling would. He had his toys. I would play with him. But I knew that he had limitations. So I was very gentle with him and growing up with him. And so I learned a lot of compassion through that. And I learned a lot about how to accept other kids and other people who are different from a normal person who does not have disabilities would be. I also remember from a very young age just getting frustrated and really angry because I knew our family couldn't do what other families could. We weren't going on some of those vacations that other families would because my brother couldn't participate. We spent some vacations going to Washington, D.C. to visit the National Institutes of Health. So one of my parents would take me around D.C. while the other stayed with my brother in the hospital. And that was a normal vacation to me. But I still had fun. I had a good time, and I have really good memories running around the halls of the hospital there. I also remember as a young kid really feeling a very strong sense of protection and responsibility for my brother. Even though he's 10 years older than me and I have another brother who's also older, my other brother also has a lot of issues. So even though I was the youngest, I felt like I had to be the oldest and I had to step up and really watch out for my brothers and be the lead of the family. And so I would try to help out my parents from a very young age. We also had full-time care from the state. We had an aide who would come into the home and help take care of my brother. So from a very young age, I would also just got really used to having a lot of strangers in our house, whether that was the aide coming in to help with my brother or the physical therapist or different people who would come in to help. But I also remember trying to keep an eye on them because I didn't know if I trusted them. I didn't know if they would take care of my brother in a way that I saw fit. So I remember trying to help them, like feed him the correct way, take him to the bathroom when his diaper needed changed, or remind them that he needed his medication administered. And I think I was doing that from as young as like 8 or 9. So because of the issues that my brothers had, I felt really strongly that I didn't want my parents to have any burden coming from me. So that drove me to have a lot of perfectionism. So I tried really hard in school to be a really good student. I never acted out or had any behavior issues just because I didn't want to add any stress onto my parents. And so I always got the feedback that I was a little bit overly mature for my age, but I think that's pretty normal for all GM1 siblings. Can we go to the next slide, please? yeah, and so the experience has changed as I've gotten older. I'm now an adult. I've moved out of the home. I've gone to college, and I'm in my full-time career. And it's been challenging because I had to move out of Oklahoma, where I'm from, to get a good job. There's a lot more opportunities for me out of the state. And so I now live in Washington, which is really, really far from Oklahoma, really far from my family. So I feel a lot of challenge there of being able to be there for my family in times of need, but I also know that I need to pursue what's important for me in my life. So it's hard for me to figure out the balance between responsibility and family as an adult. And I also feel a lot of guilt because I'm not there because my parents are stressed and I'm not always there to help. And I also know that my brother won't live as long as a normal brother will. And I worry that I'm not there just to experience those happy life moments with him. But I will say Jorgen still loves me extremely dearly. I'll FaceTime him a lot. And I don't think he fully understands what a FaceTime is, but he does light up when he sees me on the phone. I'm his favorite person in the world. He talks about me every day, even when I'm not there. And so when I go home, I just really cherish those times we get to spend together. I'll read him books. I'll take him to the pumpkin patch. You can see in the bottom left corner, that's me helping him walk. He can walk if he has a little bit of a cyst. So I still get to have that sibling relationship with him as an adult. It's just changing through the years. I would say it's a sibling, but it's also a little bit of a parent, but I still tease him a lot like a sibling would. I kind of give him a hard time and he laughs. So we're still siblings in that way. I do see a comment that someone can't see the slides. Also, I became a legal guardian of Jorgen when I was 21. So we did that in case something were to happen to my parents, and I would then have legal custody of my brother. So that was a huge responsibility for me to take on. But I also just know, as much as I love my brother and I want the best for his care, that I was willing to do that. So yeah, that's a little bit of my experience as a sibling of someone growing up with GM1. I really, really love my brother to pieces. I think he's the best person in the world, and he's just a light to everyone he meets. And I don't think I would be the person I am today without him. So I'm really grateful for all that he's taught me.
Thank you so much, Katrina. That was so lovely and awesome, and such a great segue to what I'm going to talk about, which is kind of a broader picture of the sibling experience. My name is Emily Hall, and I'm the director of the Sibling Support Project. Could we go to the next slide, please? Thank you. The Sibling Support Project is the first national program in the U.S. dedicated entirely to supporting siblings of people with developmental health and mental health concerns. We are a proud program of Kindring in Washington State, which is Washington State's largest neurodevelopmental center. And we're really proud of the work we do to support siblings like Katrina, who have a little bit maybe of a different experience than people who have siblings who don't have some kind of health concern. Next slide, please. Just to give us a little background on what we do, how we support siblings, we have a number of books written largely not only for siblings, but by siblings. We also run these really fun things called Sib Shops. If you don't know about Sib Shops and you have a school-aged child who has a sibling with a diagnosis, please check out our website to see if there's a Sib Shop near you. Sib Shops have been around since 1982, and they provide opportunities for school-aged siblings to receive information and support in a really fun, highly recreational setting. We also have some online communities for siblings through Facebook. You can find out more about them on our website. Next slide, please. I really want to dive into sort of why it's important to support siblings and why we're so grateful that all of you are here today to learn more about that. If you could just advance the bullets, please. You could just let them all go at the same time. So what we know from the research is that sibling experiences really parallel parents' experiences. And you heard Katrina talk about her experience growing up, kind of being like a third parent, right? Watching the aides to make sure that they're feeding her brother correctly, helping with diapering and all those kinds of things. We know that siblings' experiences in terms of needing information about the disability or the disorder, in terms of maybe feeling a little bit isolated from their peers. We know that these and so many more are the ways that the sibling experience parallels the parent experience. We also know that siblings are lifespan issues. I love that Katrina talked about her experience as a child and how that experience has kind of evolved and changed over time into adulthood. These brothers and sisters, you know what? We are in the lives of our siblings longer than anyone. Probably, right? Definitely longer than any service provider, probably longer even than parents in many cases. And so sibling issues are lifespan issues, and we want to consider the needs of siblings across the lifespan. Siblings also spend a great deal of time together, particularly as children. Katrina has told you about her experience growing up with her brother. We know that over the course of the lifespan, siblings spend a great deal of time together. We also know that that is quality time, that as much as I'm a big fan of inclusion, we know that there's no classmate in an inclusive classroom who will have as great an impact on the social development of a child with a disability or a diagnosis as his or her siblings, right? Siblings are 24/7 social models. And then finally, what we are really recognizing, I think, across the board as families, as providers, even government is recognizing this, that siblings really are becoming the next generation of caregivers and stepping in later in life when parents are no longer able to care for their children. Next slide, please. I want to this is going to be a super fast tip of the iceberg. When we think about unique concerns for siblings, I just wanted to kind of put these out there to give you kind of an overview of some of the things that, some of the themes that come up when we look at the sibling research. And Katrina talked so much about so many of these things, especially the increased caregiving, right? The guilt. We could talk a lot about sibling guilt. Talking about siblings without talking about guilt is like talking about the ocean without mentioning water, right? They kind of just go hand in hand. And you talked so eloquently about, even as an adult feeling guilty for pursuing your own life, right? For pursuing your own choices and dreams. I talked about that isolation, the resentment. We could sure do a whole session about resentment. When family life seems to revolve around one person, it's really easy to understand why a sibling might feel resentful about that, right? Which leads us back to guilt. It's the gift that keeps on giving, that cycle, right? Katrina spoke so eloquently about the pressure to achieve, this idea that, I have to be perfect, right? I don't want to tip the scales anymore in the direction that's going to add to the perceived burden of my parents. Very, very common. And then, of course, the future concerns, right? For our lives and our families' lives. Next slide, please. Just as parents will tell us that, maybe this wasn't our first plan A, there have been a lot of silver linings to the experience of having a rare child. Siblings feel the same way. And Katrina, you talked about so many of these as well. This idea that having your brother really has helped develop tolerance and compassion, right? Certainly you spoke about how you've been your brother's advocate over the years. And the way you speak about your experience definitely speaks to the insight and maturity that you and many other siblings have. The humor, right? I think most of us who have a family member with some kind of diagnosis or some kind of disability, I think we'd agree that we have a unique, if not maybe slightly twisted sense of humor, right? I think Katrina's agreeing with me on that one. It's like, you know, sink or swim, laugh or cry. We're going to choose, we're going to swim and we're going to laugh, right? Vocation, many of us find our way into helping professions. And I love these last 4 bullets because Katrina, everything you talked about with your brother speaks to these 4 points: the pride you take in your relationship with him, the inspiration, right? That is modeled by his resilience and grit, your appreciation of your relationship with him, and clearly your loyalty. These are opportunities that you and many other siblings cultivate over the years. And last but not least, I think we've got one last slide. I wanted to share this with you because it's a quote from a sibling who is in one of our Facebook groups, our SibNet group, which is for adult siblings. And I want to share it with you because it really speaks to the power of sibling support and connecting siblings with one another. She writes, "For the first time in my life, I have found this home. It is one I've needed for a very long time. I am no closer to, quote, fixing my brother or taking away my parents' grief. And I am still really scared about the future and not about how the world treats my brother. But I kind of feel like we are this group of quiet warriors holding each other up, pushing each other forward. Thank you all." So thank you all. And David, I think you're gonna take it away and close us out for the day.
Thank you, Emily and Katrina. I really enjoyed both of your presentations, and I want to first thank the Curie GM1 Foundation and Christine in particular for including this topic during the very packed and interesting day of the conference, because I think siblings often, one of the themes is siblings often don't get the attention that, healthy siblings don't get the attention that their sibling who's living with a long-term illness gets. And I think that sometimes becomes an issue. And by including this topic, the healthy siblings are addressed. I also want to appreciate, Emily, you and your organization providing opportunities for SIBs to meet each other, because as Katrina said, one thing about living with a sibling with a rare disease, a neurodevelopmental disorder, a long-term illness, is that your life is really different, your family life is different, and there's a feeling of difference from your peers. It doesn't mean that you don't have good peer relationships, but what a relief it must be for the SIBs to meet each other and compare notes, and then all of a sudden find a peer group where, oh, you're living this life too. And I guess I want to address my comments to parents, and I'm making the assumption that there are many parents online today. So the first is, one has to be very careful when you're talking to experts, and we're talking to experts because you are the experts on family life, living with GM1 or perhaps another rare disease. So you know your children better than anyone else, and your instincts and your knowledge about your children should be your guide. There is no greater expert than you, and I would be cautious about anyone who says that they know more than you about this. And you know as a parent that your greatest tool is listening to your children, and that is really step number 1 through a million about addressing issues of siblings and family life, living with a challenging long-term illness. I also want to make the point that I've reviewed the literature. I guess I didn't introduce myself very well, but I'm a psychologist. I work in patient advocacy for a biotech. We do not have a program in GM1 at this time. So I reviewed what has been written about siblings of children with long-term illness. And there's not been enough attention and research. That could be another theme, is insufficient attention at times. But there is absolutely no evidence that that being the well sibling of a child with a long-term rare neurodevelopmental illness causes any harm. And I think you just heard from 2 siblings that demonstrate that really well. that there can be challenges. There are challenges. There can be resentment. There can be a lot to do, but none of those things cause harm to children. Rather, I think, as Katrina expressed so beautifully, that siblings come to understand that families who learn how to work together overcome great obstacles. And that is such an important life lesson and helps siblings overcome great obstacles in other areas of their life as well. They often do seem mature because they become mature because they're often given more responsibility or see more about the challenges that life presents much earlier in their life than do other children. And, you know, one of the problems of modern family life in the United States is that we tend to protect our children from knowledge about adversity and knowledge about challenges, and then somehow we expect them as adults to know how to address it, and they may not. So most parents and families are aware that the child with the illness, with the symptoms of the illness, gets a disproportionate amount of attention in the family. And, you know, there's no way around that. They need attention to get through every single day. And so, again, that is not harmful, but I think most families already work to try to overcome the imbalance in different ways. One of which I think Katrina said, when her brother Jorgen was going to NIH, she got to go to DC and visit the various places with one of her parents. So that was a way to kind of compensate for that. I had a friend whose sister was ill for many, many years, and she has recollections of being left in the waiting room of large hospitals for endless periods of time. I think that's an outlier, and I think most families, certainly ones connected to organizations like this or who've worked together to retain family life, don't do that anymore. And also hospitals are much more welcoming to children. You know, I think there are some cautions, and I think the caution is about not giving a well sibling responsibility beyond their developmental level. And that might be being asked to stay with their sibling alone at home and being responsible when they may be not old enough to stay alone at home. And, you know, I think we watch out with all children to match the growing responsibility we give them to their abilities to handle it. So, and I think Katrina described, you know, she helps to care for Jorgen in many ways. And I didn't hear any complaint there. I think that is, you love each other and you want to care. And also the kids recognize that the parents have a lot to do and usually I think want to take part in that. I want to address talking to children about, talking to the well sibling about the rare disease. And I'll just use rare disease in general. I am definitely not an expert in GM1, and I don't want to get too specific. But Katrina was born into this life, but I think, and you can either Katrina or Emily interrupt me at any time, but at some point, you know, this is what life is like, but at some point kids start asking a lot of questions. And I'm going to guess, Katrina, you asked, well, why is Jorgen the way he is? He's different than other kids' brothers, for example, and what's going on here? And if the age difference were reversed, or if the kids are close in age, kids are likely to say, well, I think what's my brother or sister seems to be different, or that he's described as being ill and having an illness. So what is that and what causes that? And I personally am a big advocate about being honest and straightforward with children and not trying to hide Emily, your talk was a bit more general, so I'm referring to Katrina more, but I think even the lifespan of a child got discussed in your family, and you became, I'm going to guess, when you felt ready to ask what your brother's future would be like. And the important thing for families to remember here is that children develop not only physically, but they develop cognitively, and their understanding about the way the body works and about how illness happens develops over time, and probably a little bit more slowly than we expect. So there's research, and if anyone is interested in this, please contact the organization and I'd be happy to send information. But children don't really perceive what's going on inside the body under the skin until they're close to the age of 7. So when we try to give complicated descriptions or explanations of illness that have to do with organs, that have to do with something going on inside the body, it is very difficult for kids to understand. They're just not there yet. And so and they tend to see the causes of illness outside the body. So if you ask a child, if a child said, you ask a child, how do you get a cold? They'll say, oh, well, the sun gave me a cold when they're very little. And later on, they'll say, maybe I was outside and I didn't have a coat on. So, but none of it has to do with, viruses in the nasal passages or the immune system, that all of those things come much later in life. So without going through every stage of development, I think the important point is to match the explanation, not just about the medical part of the condition, but of the other parts to the cognitive development of your child. So you don't need to make it too simple, obviously, but not to have an explanation of the brain to a child who hasn't really thought about brains or isn't going to think about brains for a long time, for example. And again, you're experts, so you know what they're talking about and you know what their questions are. And I think there's a match there. So your kids listen to you, they're like experts on their parents and on their siblings. So they listen in to what causes illness. And the first one is contagion. And of course, we've just been living through a highly contagious period. So there's a lot of discussion about contagion. And, so they say, well, will I catch it from, my brother or my sister? But, will I catch GM1? And, I think there's a pretty clear answer to that. And the answer is you do catch some things like COVID or a cold, but you don't catch this kind of condition. And sometimes it's like, it's about, did the child do something to cause the illness, like going outside without a coat? And I think the answer is pretty clear that that's not the case. I don't need to tell you. And then in some cases, and when kids get older, they hear that some illnesses can be inherited. And depending on the condition, that could be a much more complicated response. And I think, again, you have to be very cautious to make sure that you are going at the level of your child's perceptions. So this is another very large topic that I wanted to bring it up because I think that both the involvement of your child in taking responsibility in the family and the explanations for the condition are very much age-related. So I think we're just about ready to stop. I just want to thank my co-panelists and see if you want to use the last 2 minutes for any comments of your own. And it's just a great pleasure to participate in this program. So thank you.
Katrina and Emily, did you have any additional comments? I do have one.
Just to echo David's thanks. And David, so much of your talk reminded me of the expression that children are excellent observers, but not always the best interpreters, right? And I think that's so important to remember when it comes to sharing information about the disease. So anyway, thank you all for having us. And thank you, Katrina and David.
Yes, thank you all.
I do have one question. Are you three able to stay until the question and answer section at the end? Awesome. So I will hold that question until the question and answer section. And then this next topic, GM1 family clinical trial experiences, I'm actually one of the panelists and a part of. So Dawn is jumping on to actually moderate this section for us. So Dawn, I'll let you, actually, do you want us to each introduce ourselves quickly and then we can go into questions?
Yeah, I think if you can quickly introduce yourself, that would definitely be helpful. And this is a super exciting topic. I mean, we're fortunate to be in the position to have clinical trials ongoing, and then to be able to share with the families the experience that we've had. One quick reminder that I did want to sort of make is, this is not a confidential forum. This is not a HIPAA compliant platform. And so please, and the clinical studies are all ongoing. They remain blinded. So just a reminder to please don't disclose the trial that you've participated in or the drug that your child has received. And just, happy to hear, and everyone share their experience, what it's like to be in a clinical trial. So I'll pause there and then maybe Kylie, hand it back to you.
Mm-hmm. Yeah, again, my name is Kylie Harrison, and I've got 2 kiddos with juvenile GM1, and we are grateful that we have been able to participate in one of the clinical trials available. Very grateful that that was even an opportunity for us, and have to say thank you to the foundation and Christine and all of the work that has happened up until this point for us to be able to participate. I think for us, the trial has been, it's been a whirlwind of everything. You feel so grateful to participate. You have excitement that this could change your children's lives. You're still scared. You're still worried. There's just so many emotions when participating. Our experience, I think, has been, for the most part, very good. We have been treated wonderfully by the staff. Our children, have gone through a lot. And I think that's one of the things that I would tell parents, who are debating about trials and if they have choices and participating is it is a lot and it's a huge commitment. But of course, we're doing it for them and for research and development of a treatment for all GM1 kiddos. So while it's super challenging on our family, we've continued to want to participate in and are grateful for that. And I guess I will pass it off. David, do you want to?
Yeah.
I'm in.
Hey, everybody. My name is David Law. First of all, thank you so much to the foundation, to Christine and everyone for organizing this great forum. So me and my wife, Vina, were the parents of Violet, who is our daughter affected by late infantile GM1. And we were very fortunate to participate in a trial. I'll preface this by saying that me and my wife are in the medical field. So we had some sort of knowledge of the trial nature and how things, what to expect, I guess, on paper, I guess. But certainly going through it is a whole different experience. Everything from the pre-laboratory workup to the interviews to waiting to see if you ever get in. It's funny, like we're both in healthcare, me and my wife, when we both applied to medical school and residency, and this kind of felt like the same thing, almost as you apply a bunch of people, you email people, and you wait on for weeks and on end for something to happen. But certainly we are very happy to be part of this, not just for Violet, just to be able to contribute to the community in this way. I think a lot of it for us has been just kind of like getting a realistic sense of what, I guess, a cure looks like, right? I think initially when all this happened, we were just so caught up with, you gotta find a cure, gotta jump on it real quickly. the reality is that, well, you develop therapies, and I think we are now more well versed in the fact that these forums provide a way to develop these therapies that can be part of maybe a number of treatment modalities in the future, and just so some treatment options in general, just for these families that have no other recourse. So, I think definitely going through the initial steps was a little, harrowing. We definitely had to go through the labs and, the interviews and a bunch of other things along the way and a couple of procedures. But overall, we are very happy with it. And, we do encourage, other families to come forward and hopefully be able to participate as well. So with that, I think we'll pass it on. I think who else is on here? Maria, would you like to go next?
Sure. Hi, I'm Maria Bacon. I have 2 boys who are affected by juvenile GM1. And my younger son was treated in one of the trials coming up on 3 years ago. So we're several years out now. And I think even though we had done natural history studies before participating in the trial, so we had a little bit of an idea of how intense it would be, it was just way more intense than we could have even imagined going in. I don't think you can be prepared for how much it is on both the child and the family in general. But overall, we are really thankful that he has had the opportunity to participate in this trial. Martha?
Okay. My name is Martha Louderback, and I am Bentley Louderback's grandma. Ours was kind of the same situation. We were just so thankful for the trials and the opportunity to be part of them. It was much more intense than kind of what we anticipated maybe. But it was, I feel like we are a year out from the trial, and I feel like it was well worth doing. We've had great interaction with the site and the company and the doctors and staff. Everyone that has took care of Bentley have been great, and we appreciate the opportunity to be able to be a part of, well, making history. And I believe that it's just one more step closer to the cure. And we're just grateful to be able to be a part of that and have such a support group in the GM1 Foundation, family, and meeting other parents, other grandparents of GM1 kids has helped so much, because when you do get the diagnosis and you realize how rare it is, you really do feel alone and kind of just out in the middle of the ocean by yourself. But then, we got in contact with the GM1 Foundation and discovered that we're not alone. It is a small ship, but we are not alone. And we have, we've really appreciated that. And I just think that it's a great cause to continue on with and see where we go from here.
I see that. Thank you, Martha.
Thank you.
Dr. Hastings has joined. Would you mind we just are doing some quick intros. If you wouldn't mind introducing yourself.
Sure. Thank you. So I'm Caroline Hastings. I'm a pediatric hematologist and oncologist at Children's Hospital in Oakland, and I've been running clinical trials for my entire career for the last few decades. And more recently, I'm one of the sites for one of the GM1 gene therapy trials. And so I do know, of course, a number of you and a number of other parents, and I'm very happy to be here and to help answer any questions.
So thanks, everybody. I thought maybe we could lead off with some sort of obvious questions that families might have around participating in a clinical trial. So one question would be, what is the time commitment and how do you manage that with your everyday, day-to-day lives.
I'll go ahead and start, I guess. So in our trial is currently ongoing, and frankly, you know, we don't do we actually don't follow up too much. We follow maybe once every few months at this point, and have a meeting with our neurologist and do a couple of blood tests and such. So really on a day-to-day, you know, we get a lot of time to spend on violence therapies and, you know, going to preschool and other things too. And at this point, at this level of our trial, we're not checking in that much except for every couple of months. Now, initially, there was a big time commitment leading up to that. We're talking about lab tests, talking about we had a lumbar puncture, we had to do an overnight observation in the hospital, EEGs. So those months leading up to it were very time intensive. And I think once that petered off and things started, we're definitely at the tail end of it now, more observing and seeing how it went. We have more time to focus on taking care of Violet.
Any other comments?
I'll add to that and I agree, the initial time frame, we were very blessed that I was able to just not work and kind of take over Bentley's care. We spent about 2 months out in California for the first initial testing. And like David said, the lumbar punctures, MRIs, labs, all of that, and the testing for the trial before treatment. And then about a month after treatment that we stayed. And we follow up every 3 months with the site in California. We travel back there. So time commitment, I will say it is a time commitment, but at the same time, I wouldn't do it any different.
And Maria, were you going to say something?
Sure. Yeah, ours sounds about similar to the other two. The initial time commitment is just huge. It was just over 2 months for us spent on site in the area with a week of initial, testing, doing everything under the sun, MRIs, developmental testing, just blood tests. And then after that, weekly check-ins, immune suppression, and then the treatment, and then follow up afterwards, just being at the site and checking in regularly and just being away from home for that initial time period was, a huge commitment. And then that first year we had to travel back every few months. So during that first year and a half, the travel and spending, about a week on site every time was very intense, but now we're down to just returning once a year. So the time commitment has reduced dramatically at this point, and it doesn't feel so much of a burden for, our family anymore like it did initially.
And in terms of for families, and again, to remind everyone, we'll have Q&A at the end of this session where people can ask more questions. We just wanted to start with this initial dialogue. For families who are considering participating in a clinical trial, any words of advice you might have for them as they make that decision?
I'll start off, I guess. So, I think in our situations with this kind of disease, of course, being in a trial is obviously like the most viable option when there's no other treatments really available aside from supportive care. That being said, I definitely think you should make sure everything's checks out with the trial. I mean, right now there's only so few trials really available, and you can see all those through the NIH website. But I think some of the bigger question is about just kind of finding out what is open. And I think Christine and the GM1 Foundation have done a really great job about keeping us in the loop of what trials are actively open, recruiting, and ongoing. So that's a great way to keep your ear to the ground, and see what's going on. And at least my second point that I want to say is that, I think the bigger question now is like, well, how do you find a trial? How do you get in contact with the right people and actually even get the responses you need? And, I wish it was like just filling out a form on a website and like waiting for someone to get back to you. And, you know, ideal world, that would be great. Yeah. But really sometimes it takes down to like, you know, calling the right people, knocking on doors, you know, and just being persistent. You know, frankly, in our trial, I relied to some old colleagues from residency who happened to be in the same facility, and they were able to advocate for me. And then I eventually got a call back, you know. I wasn't going to stop trying regardless, but that I think it speaks to the rigor that you need to pursue and the follow-up you need to make this thing go through.
Yes, that's definitely, I agree with David on that, is you have to be your own advocate. We were very lucky early on to meet Christine and get in touch with her. And she sent me all the, you know, the information on the trials that were out there, and links, connections to be able to contact the different trials. But you definitely, you can't sit back and wait for, you know, them to look for you. You've got to be your own advocate, your child's advocate, and really pursue it, because it is such a rare disease and the community is small. So it is easy for those things to fall through the cracks and you not be able to really find them. And so having a source like the GM1 Foundation is priceless, and they help. I know that at any point in time that I can pick up the phone and call or message Christine or the GM1 Foundation, and I'll get a response. And so that you do have resources and help out there. So my biggest word of advice would be utilize that, and just, you kind of have to be relentless a little bit.
Yeah.
I think I would chime in to say that, you know, even once you're accepted into the trial, to continue to advocate for your children, and possibly, you know, like we've asked for different changes and accommodations based on our kids and the differences. You know, they have ability, just like any medical situation that you're in with your child, you know, they can adjust some things to your child. So like Kennedy hates going under with a mask, right? So, you know, they've adjusted to that, and they put her under with an IV instead, because that's her. So just continuing to advocate and voice for your child during those procedures, if you are accepted into a trial, and continuing to have that open conversation, and also telling them what you can and can't do as a family. I think that's the other thing I've learned, is I haven't been afraid to say, like, no, we can't go that week, or no, I need to bring my infant. So can we do this? Can we do that? You know, just advocating for your whole family as well, in, you know, making it doable for your family, because they are going to help. They want you to participate. They want the data. So continuing to be a voice.
There are some questions that have come in from the audience. So I know we have some more time at the end, but I thought we would use this time. So one question was, as a parent of a young child, I'm nervous to have my child take immunosuppressants as part of a trial. Did this trigger concerns, and how did you navigate and feel okay with that? Maybe if one of you could take a stab at answering that, and then we can go to some of the other questions that are coming in.
Yeah, I'll jump in real quick too, just because, I mean, we did it during COVID. So there was extreme worry, and at the same token, we were also blessed that it was COVID, because everybody was extremely careful and wearing masks and whatnot. But in at least our clinical trial, you know, they weren't immunosuppressed to the point where I felt like it was super dangerous or, you know, super worrisome. So for us, that wasn't, you know, a huge issue.
Okay.
Yeah, I agree with Kylie. The immune suppression was going in a concern, but it ended up not being a huge ordeal for us. My son never had any sort of reaction to the immune suppression. And even, you know, coming home and him still being on it for months afterwards, we never had any problems at home. And we have a busy household. He had a couple colds while he was taking the meds, and it never became a large issue for the degree of suppression used in the trial.
Great, thanks. There's two more questions I think we can tackle before we go on to the next speaker. What factors would make you select one clinical trial to join versus another?
If you get in or not, I mean, in an ideal world, we'd have our choice, but frankly, in the limited situation we have, is I think you literally reach out to every situation possible and see what you can get into or what you're even eligible for. I think that was a big wake-up call to us, was that when we started applying, we thought, oh, we'll just apply and such a rare disease, be fine. Well, then the wake-up call is that you start to realize there's a bunch of other families with the same rare disease, and there's different levels of functionality, and there's a whole other process that you may not even be aware of. So I think that reality check was big for us, and to us realizing what the atmosphere was like, and just keeping, more importantly, was keeping the connections for future trials too, was very important.
Yeah, that was for us too, that I applied for all of them I found, and the ones, all the ones that Christine sent me the links to and everything. And I remember meeting with our doctor in California over Zoom, and after our initial meeting, I told him, I said, I just want you to know I've applied for the other trial as well, and if they call first, I'll accept, because and he said absolutely. So it was the community, the different trials, they are understanding of that as well, that you just apply for them all, and then when you get calls, if you're lucky enough to get calls, then you make that choice at that time. But I strongly suggest apply for what's out there.
Great. And we're a little bit over, but I think we can answer this last question, which I think is also really important for people who are considering enrolling in a trial. When you get accepted into a clinical trial, what kind of preparation does the site do to help you get ready? Not specifically the medical test, but everything around it, like the travel, the procedures, the whole process of participating in the trial. What sort of support does the site and the company provide?
In our particular, I'm sorry, go ahead.
Well, I could probably talk about that a little bit, but in general, the site should really be providing all of the accommodations and travel. None of that should have to be burdensome for the family. They have already enough on their plate. And so we try to, we work with like a third party person to make all the arrangements that would be the most comfortable and expeditious for the family. And so, it just depends on where they live. It could be renting a car for them. It could be flying them in and just making sure they get to a hotel that's close by and that's comfortable. Well, we haven't always used hotels. We've used more like apartments because that feels more comfortable for families. And sometimes families need to travel with more than one parent, especially if there are other young children. And so we do make accommodations for that as well. And then, working with the site, we try to be very aware of what the, all the difficulties, in getting a child here and what their special needs may be, medications, oxygen, et cetera. And so we have to prearrange for those things as well. And we do. And sometimes we've even had to arrange for, to have a nurse checking in on them periodically and to have supplies delivered ahead of time so that we can be prepared and it can be a safe transition.
Great, thank you, Dr. Hastings. Any other last quick comments on the last question from any of the families?
I know for us, our trial, we have a travel coordinator that is exceptional. She's in contact with me constantly. Is there anything else we can do? If I have any needs or wants, I contact her, and they go above and beyond to try to accommodate what we need. And
that travel coordinator was provided to you?
Yes. Yeah.
Great. Okay, I think we should pause because we do have some more time at the end for questions and we're a little bit over. So I'd like to turn to Vanessa.
Hi, great, thank you. My name is Vanessa Rangel Miller. I'm a board member for the Cure GM1 Foundation. Over the years of working with the community, it's been very exciting to see how far research and interest has progressed. And I'd also like to thank the previous panelists and families for sharing their experiences on participating in trials. So we look forward to their questions coming up. Why don't we jump to the next slide? There we go, thank you. So we've heard throughout the sessions today about many past and ongoing efforts that aim to learn from the experiences of those with GM1 and their families. And these efforts really have the potential to accelerate the development of new treatments. But you can see from this list and from what you've heard today, it's quite a lot. It relies on the engagement of the partners shown here and the participation of families. In fact, many of the families listening to this webinar now or the recorded version may have participated in one or more of these programs. So if we jump to the next slide. Thanks. Apologies for the strange layout here. It looks like the file might have adapted itself a little bit. But while the programs in the prior slides seem similar, they do serve different and unique purposes. And they're all centered around those living with GM1. And rather than spending time describing each, you've heard some of this already, we can look at themes as we talk about how you can take action. So natural history studies, just quickly, may look at the course of disease over a person's lifetime. And these are studies that, as Michelle shared, can collect the sort of data that would be used and compared to therapies in development to evaluate for a potential benefit and impact. Can also be used for other purposes too, though, that we may not have touched on, such as identifying standard of care or centers and sites. Medical records review, which is the next bubble, that contributes similar information from a historical medical records type perspective, and there's studies like that ongoing, while patient registries, those often look to collect the direct experience from families and individuals, more in real time, collecting direct patient report or family report information. We heard a bit about video capture studies mentioned earlier. Those collect video data of patients really doing specific at-home daily life activities to find new outcome measures. Outside of the types of assessments that would normally be done in the clinic, and to hopefully find impacts that are meaningful to families. Gene variant data, those are studies that can allow sharing of genetic and health information, and doing so in a way that protects your loved one's privacy and your privacy, but helping the community learn more about gene disease associations. And lastly, caregiver experience. We heard from Holly earlier this morning that these are studies that can explore individuals and parents and caregivers' priorities and preferences related to new therapies for GM1. If we go to the next slide, please. Thanks. So we also heard several times about the importance of biomarkers and newborn screening efforts to really help recognize the greatest potential benefit to treatments that are in development and coming forward. So here are two example projects that are either planned or underway that address newborn screening. The first is a newborn screening biomarker study from the University of Washington. The second is inclusion of GM1 in an ongoing newborn screening pilot study in New York State called Screen Plus. This pilot strives to expand newborn screening to include over 14 new additional rare diseases beyond those conditions that are already screened for by current newborn screening. If we jump to the next slide, please. Thanks. So, from this, there's a number of ongoing projects, a number of studies that we've heard about, and it's really important to remember the critical role that families can play in helping to drive all of this forward. Many of today's speakers have actually talked about those steps, the things that you can do. You can join studies that collect data to understand what it means to live with GM1, to understand the similarities and differences between the subtypes. You can contribute information, data, experiences that look for associations between genes, gene variants, so genotypes, if you've heard that, and GM1, or phenotypes, if you've heard genotype-phenotype associations. That's what it means. You can recognize the journey that individuals experience and help to identify and barriers that are really barriers for diagnosis, for an earlier diagnosis and for access to earlier treatment. You can join studies that help support the development of things like biomarkers, help support development of earlier newborn screening opportunities and earlier diagnosis. Sharing your thoughts on unmet needs, on desired outcomes, on risks and benefits is critically important. And it's important to hear from families on those when considering new therapies and development. And, importantly, we heard today quite a bit about the importance of natural history. In SMA, from our earlier speaker, we heard about the importance there. And you can participate in studies that collect this sort of information for potential use as comparator data sets for clinical studies. As part of this and other studies, you can support access to data sharing for future studies by sharing your information, but also by advocating that data be available. So if we jump to the next slide, I really want to spend a moment on how you can take action. So I'll pause and have you, I'll talk through each of these. There's some animation here. So what can you do to take action? First, donate and fundraise. Research is costly, it's time consuming, but funding research is what drives it forward. Next. Thanks. You can help raise awareness. This is another big one, easier to do. Raise awareness of GM1 and specific projects on social media. Don't worry if you don't have millions of followers. Every communication helps. So whatever you can do will help raise collective awareness. Next. You can advocate for legislation, and this can include advocating for access to earlier treatment, earlier therapies, earlier screening and newborn screening, earlier diagnosis. Next. You can join a clinical trial or a research study, and many of those have already been discussed this morning. Next. You can participate in patient and family reported studies, and these can include registries or medical history studies. Next. You can take part in data sharing projects. Sharing your data for use in future studies is key, and allowing that data to be de-identified and used again is key as well to avoid repeating studies. Next. You can take part in natural history studies, and we heard quite a bit about those this morning as well. Next. You can share your story with the community. There's a wonderful function on the website. You are the expert. We've heard that today. You are the expert. Please share your story and help us communicate what your experience and journey has been. Next. You can stay informed by subscribing to our newsletter, doing things like attending this conference. And next, there it is. Most importantly, attending this week's landmark externally led patient-focused drug development meeting. So we're very excited for that activity on Friday and look forward to the good information that will be presented at that time. So one more slide, please. There we go. Oh, I'm sorry, one more. Thanks. You can click through one more time. So with all of the projects underway, what does the future look like for GM1? And this busy diagram comes from Genomics England. It was a roadmap for genome-based newborn screening, which you've probably heard quite a bit about in the news, given various studies in the US that are tackling that, as well as similar studies internationally. However, I felt like this really captured many of the milestones and landmarks that were happening for GM1, so I've renamed it. Together, the community that we've talked with today of clinicians, scientists, caregivers, patients, parents, families, we're collectively tackling all of these landmarks. And it's really your engagement and your advocacy that are critical to help shorten this journey. As Phil Riley shared at the very first session, it's the experiences of those with GM1 and their families that are more valuable than you realize. And you can really take action using those experiences to help accelerate this journey and advance everything that you see on this screen here. So you can advance forward one more. And I just want to say thank you. I will hold questions for the end, but I appreciate any questions from folks who really want to take action and understand what they can do next. So thank you.
Thanks, Vanessa. And then David is ready to go. So I'll let you jump into your next talk.
Thank you so much, Kylie. Hey everyone. So thank you so much for this this time to speak a bit about our journey, our experiences last year with our baby daughter Violet. Were there slides? I don't see my slides. There we go. Beautiful, beautiful. All right. So we're going to spend a few minutes here and just going to go over a bit about what we've been through as a family the last couple of months since we got our diagnosis. Well, God, more than a month now. It's been more than a year. And so that is a picture of our beautiful daughter, Violet. This taken, she was probably about 9 or 10 months old. This is before we knew we got anything, knew anything was wrong. And it was taken, you know, just at home with us. So next slide, please. So before we jump into all of that, let's take a step back and tell a little story about us. Next slide. There we go. Okay, so a little bit about us. So this is me and my wife, Vina. And the unique story about me and Vina is that we fall in that intersection between being both rare disease parents and healthcare professionals. We're both physicians, and we both met during a training program in University of Colorado in Denver. There's a picture of me when I was a couple pounds skinnier, and there's a picture of my wife reminding everyone to get their annual flu shot, which is very important this year. Please jump on that. So in the meantime, we both practice in Southern California. And then we are also very active in Southern California. I also am a musician, and you can see that's one of our engagement photos there, me playing the saxophone to my wife by the LACMA lights, and a picture from our wedding party that I wanted to include on there because a lot of part of this story is about community, and if nothing else, what our experience with VITA has taught us is the value of our community and how they've all come through for us this last year. Next slide. And of course, here's the change of our lives and the beautiful light of our lives. Miss Violet Aurora Season Law, born June 11th, 2019. That was a shot right from the OR. It was a C-section, and luckily there was no other complications or anything. And she was born beautifully without any issues. Next slide. The first couple of months of our newborn's path were just like any other newborns. Sorry about that. It was like any other newborns situation. We had parents come by, we had a lot of family coming by, we had a lot of support and a lot of good visits from people, and everything for the time seemed normal. This was, of course, end of 2019 going into 2020. Next slide. And of course, the world changes. As we are raising our child, the world changes with the beginning of the pandemic in 2020. And I was a hospital physician, and I still am. And during this time, it was very scary. And this is a quick little plug and shout out to our frontline workers who no doubt have coming through this ordeal and have a lot of issues coming from it. Well, you know, I can say coming through the pandemic, one of the things that I wanted to do more than anything in the world was to sit back and be over the pandemic and raise our child. However, as the pandemic neared, in the waning months of the pandemic, we started to notice that Violet was starting to have more developmental issues, and that showed us hypotonia, lack of motor functions, and lack of achieving certain milestones. My wife, Vina, is a developmental pediatrician, so we were able to identify these things rather early. And based on that, we pursued a quick, a diagnostic odyssey, which then revealed us to diagnosis of GM1. Next slide. This is a post from the day that I posted about Violet's diagnosis online. And I think this I feel like a common thread from a lot of the parents on this panel is that we all have this day, you know, the day you got the diagnosis, the day that you told the world, whatever it was, that landmark day. And for us, this was that day. And it came shortly before her second birthday, you know, and it was so heartbreaking. I'm sure all you've seen, all these hopes and dreams and all these things suddenly being put to come to a halt, and suddenly a change of your overall plan and life view for her just kind of comes at once. And certainly we went through a lot of the same emotions and a lot of the turmoils and ordeals that everyone already went through. But in the time that I have, I don't want to spend that focusing on those times, but in that the amazing response that happened afterwards and what propelled us into the journey that we're on now. So next slide. So one thing that was immediate was that the community that we've been so involved with and so close with all throughout California, they really came through for us. And within a few weeks, more than $100,000 was raised just for GM1 research. And that came from families, friends, people all over. Christine from Cure GM1 was able to go on the local news for us and talk about the Cure GM1 Foundation. We talked to local researchers and just got out there as much as we could and raised as much funding and awareness. I think in these early days, it probably was more of a reflex response. I think something tragic happens and, I think a lot of us grabbed to what was most, tangible. And a lot of times fundraising is a very tangible way to give back and contribute. And we certainly dove into that. And next slide. This shows a couple of the news outlets we were a part of. The Long Beach Post is very good in covering us about the story. And from that alone, so many people in the community came out and sent us nice emails and special messages and donations just for Violet's cause. And then, see on the right down below, that was me about a little more than a year ago, talking in the same chair that I'm talking from right now, but just talking to the news about how this was all just very new to us, what we were doing to try to cope and deal with it. Next slide. And then the reality of more of this disease did hit. I think this is after a couple months of disease, and Violet got her first bout with viral pneumonia. It did cause some hypoxia, and she did have to go to the hospital. It was a tougher time, but it did help that we were both had medical training and were able to deal with that. But for us, it's a bit of a wake-up call to, in the middle of all this advocacy and fundraising, by the way, you have a very real job of keeping your child healthy and keeping, taking care of her issues. And I think that's what kind of the theme of all this has been. There's been both a long-term overall picture of the Cure GM1 and advocating for cures, research, and all that. And then there's your acute day-to-day basis of taking care of the sick child. And that's what we've been doing. So next slide. Luckily, after that bout, I did recover. And I saw her a couple of days later being happy and healthy. And, every time she gets sick and she bounces back, it does remind me that despite the disease process, that there's a kid inside there and kids bounce back. Of course, some take a little more than others, but naturally her body wants to get up there and be part of the world again. So we were blessed that she came out of that situation okay. Next slide. And so where are we now? And I think this shows a little bit of some of our efforts. It became very obvious to us earlier on that everything that we can do for Violet, taking care of her and making sure her day-to-day is comfortable. We were all doing that. But, all these other energies that we had, all the other resources, how could we pool them to just really advocate for her? We have friends in the music industry, and here in Los Angeles in January, we were able to organize a Cure for Violet music fundraiser. I got to bust out some of my old piano singing chops and got with my old band. But we also had Black Joe Lewis and the Honey Bears come and perform for us. And we rent out the Mint in LA for a night. We raised thousands of dollars for GM1 research and had a great time talking to local media and people around about GM1 and everybody. And it was also a night to celebrate too. I think this was probably several months after the diagnosis, but we've been through so much sadness and coping, and there's so much bad news that this is a night to just sit there and say, and to acknowledge ourselves for, hey, you've gotten this far. Violet's nice and stable. We've done a lot for it. Let's celebrate with our friends and use this energy for a good cause. And that, I think, has spurred a lot and propelled us to really just focus a lot on advocacy and pushing ourselves out there as we pursue treatments for Violet's disease. Next slide. So this just kind of shows a little bit about how the Cure for Violet campaign has gone. This is recently in Las Vegas. One of our friends runs a music festival, but we had Cure for Violet t-shirts out there. People wore them in and out for the casinos. Really cool. And every time people would see them, they'd ask about Cure for Violet or what that's all about. And that would then lead into either a longer discussion about GM1 gangliosidosis, or more commonly, a quick exchange of websites so they could learn more about it. But upon all that, it was so heartwarming to see so many friends, and not just my close friends, even friends from the periphery that had heard about our plight just come out of nowhere and just want to advocate and care for us. And a little bit ago, we talked about the silver lining of everything. One of the biggest silver linings was everyone coming out and reaffirming these bonds with people that we had who came out to show their love for us and for Violet. Next slide. And fitness. I've never been the most athletic of people, but this did spur me to be a little more athletic for myself and for Violet too. On the left is my first half marathon in April, which was the OC half marathon. I guess I set a PR because it was my first marathon ever, but I did limp across the finish line after 3 hours and 30 minutes or something, but it was important. And as it was good that and Vina and Violet were there at the finish line to wait there for me. On the right is just from this last Sunday at the Long Beach bike tour, 26-mile ride throughout Long Beach. I'm there with the LA water symbol, whatever it is. But that was another bike ride I did wearing my Care for Violet shirt and also for GM1 research too. Next slide. So I think the point of this was to kind of show where we're at. On the left, you see a photo of us just shortly after diagnosis. We still got it together to do a birthday party for Violet in the park. And at that point, not really knowing where the road was headed. We didn't know, based on all the numbers, would she deteriorate quickly or how things would progress? Would she stabilize? We had no idea. And if you fast forward a year later here on the just a couple months ago on her third birthday, here she is stable in her cute lavender dress here with our family and friends. And, even though there's a new reality that's set in with us, this is still our family and still our lives. And I think the focus on this has been figuring out that it's not about how long she's going to live or what is going to happen in the future. There's a lot of that we can't control. But what we can control is her experience here as she's with us and all the energy that we have that are making sure that's going towards positive solutions and therapies and advocacy for her. Next slide. So, this is a collage I just wanted to show of just like this things that we've done and how much of a life we've lived this last year, going to pool parties, going to various trips. Las Vegas and Palm Springs are big favorite parts of, location that we like to go to. We dress her on Halloween. We have a great costume for her coming up soon too. She loves the pumpkin patch. And we really do focus on making sure that, every moment of her life is not spent, it was spent in comfort and in happiness and in enjoying the things that life has to offer. Next slide. And to close it off, this is the last thing to epilogue, and this, I guess we had a little spoiler surprise before, but we were lucky enough to be accepted into a clinical trial. And I think that just goes on, that testifies to, you have this idea that you get this diagnosis and you have only so many more years to live because it says, oh, average age is 4 or 5 years, and in medicine, you see it all the time. But the same advice that I tell my patients in the hospital was the same one that I found myself reflecting on when going through all this, was that it is just a number, right? Yes, it is a number and there's a lot of evidence behind it, but at the same time, how much of that can you control, right? How many of that is due to avoidable illnesses or the lack of therapies or whatever else, right? How much of that can you really put into your own hands and advocate for and make your own reality? And I think that's what we've been pushing for the most. So again, we thank everybody for their support and their help through all of this. It's now a year plus past her diagnosis. Violet is just started preschool. We are in a clinical trial. Life is going forward. We're going to go to Disneyland in a couple of weeks, check out the Boo Bash thing, and just enjoy life. So we wish all of you the best, and please reach out if you have any questions. But thank you so much, and God bless.
David, thank you so much for sharing your story, not only today, but with the world and for your advocacy efforts and fundraising efforts. We really appreciate that. And then our next speaker, Dr. McNally, I'll let you introduce yourself and take over.
Thank you, Kylie, and thank you, David, Dr. Law. My name is Tom McNally, and I am a pediatric palliative care and rehabilitation medicine doctor here in Oakland, California. And the timing of my talk couldn't have been better because I was listening to Dr. Law, to David, and tell the story of Violet and David and Vina. And I was thinking about how much what we just heard in the last 10, 15 minutes was exactly the sorts of things we would want to know as a palliative care team. And what an extraordinarily beautiful glimpse we had into this wonderful family, all the deep love they have for their child, and where they find meaning. And I heard that they find meaning in Violet being comfortable, but also in working to advocate for her to get treatment and for other kids to get treatment. And that's where, faced with a diagnosis that has a lot of uncertainty and a lot of pain and fear that could be attached to it, David and Vina took that energy and transformed it into action. And as a palliative care doctor, those are the things that I want to know. I want to know about the child and I want to know about the family so that we can help as we come alongside you in your journey of a serious illness, and particularly a serious illness that's connected to uncertainty like GM1, that we can be with you and help you navigate this as best as possible. So next slide, please. So I'm going to spend a little time talking about what pediatric palliative care is, some of the communication strategies we use, and a little bit about symptom management, and really not going to get into the weeds with that, but just give you a sense of the things that we think about in case there are worries for you. So next slide. And I have no financial disclosures. I'm not, don't have any investments in any drugs or anything like that. So again, this beautiful example that David and Vina had just given us in telling us about Violet, I'm going to ask you to think about your child and think about something about them that you'd like to share with their medical team, but you don't usually get asked. And something that's really special about them, something that that fills your heart with joy or love and delight. And if you feel comfortable with that, please add it into the chat box if you want to just say that, or if you just want to hold on to that idea. And then secondly, after you've thought about that, imagine what it would be like to share that quality or attribute with the medical team, and what would that feel like? And maybe you have done that. I hope you have. I hope you had a chance to tell the people who care for your child and for you as a family what it is that you love about your child, what is it that makes the child so special to you. But if you haven't, think about what it might mean to add that to the conversation. Because those are the things that I really want to know. I want to know about your child, and I want to know what's important to them and what's important to you. So what is pediatric palliative care? The word palliate means to cloak. And this is the sense of like to wrap around around someone a comforting blanket is the way I like to think of it. There is the medical hug, as one of my colleagues used to say. And the definition that is written here is from the WHO. That's from 1998. And I think it's important to even though it's a lot of words up here, I think it's important to pull out a few key concepts. It's active total care of the child's body, mind, and spirit. So I work with a social worker, chaplains, with a child life specialist. I work with psychologists. I work with integrative medicine specialists as well. And so we're thinking about all components of child's physical, spiritual, psychological health, and also for the parents. Ideally, palliative care begins when the illness is diagnosed and continues regardless of whether or not the child receives disease-directed treatment. And this can be a little bit confusing, especially because this is a distinction from adult palliative care. Many of you may have had parents or grandparents who have died or faced a serious illness, and they've received palliative care in the adult setting. Generally speaking, that is primarily focused on the time when a person is approaching the end of their life. For pediatric palliative care, we can follow kids for many, many years and support families and the children during that time. It's not always related to an imminent death or imminent deterioration. Health providers must think about all the aspects that I talked about before. It requires a broad multidisciplinary approach, and we use all the resources available within the hospital and the community, and can be provided in tertiary care facilities like where I work at University of California, San Francisco, in both the Oakland and Mission Bay campuses, and community health centers, and in children's homes. And I'm also one of the attending physicians for our local hospice group. It's called By the Bay Health, and the pediatric group is called By the Bay Kids. And so we provide both in-home palliative care and in-home hospice care for children. Next slide, please. So here's the plug for our particular place, but I want you to know that those of you who are in the Bay Area, Kaiser has a palliative care program, Stanford has a palliative care program. If you're in Davis, there is a program there as well. And so there are a number of institutions. I just happen to work at UCSF, and we're called the PACT team in Oakland and the IP3 team in Mission Bay. Next slide. So these are the questions when I go in to meet a family for the first time. So if I had the wonderful opportunity to spend time with David and Vina and Violet, and these are the questions that would be in the back of my mind as I went in to have our first visit together. And I would ask, as I said before, tell us about your child. What is she or he like as a person? And what do they like? What are the things that bring them joy? For families whose children are living with chronic complex conditions, we'll often ask the question, what's a good day look like? What are the things that make for a good day? And also what makes a bad day? What do the tougher days look like? And often we will say, if you're seeing that your child is having more bad days than good, that might be a time to think differently about what the goals of care are in thinking about your child. It may not be, but it's a time that we sometimes will reassess and say if a child is having more bad days than good. Second question is, what is your understanding of your child's illness? And this is really important to me, I have to say, because this is a way for me to know not only what parents get medically about it, but what story they're telling about their child and their family's life together. And that can be really important. So for example, we say, if you use the terminology that, which a lot of people use, my child is fighting against a really difficult genetic condition, then we can think about what a fight looks like. What does it take to be strong in the fight? What does it take to recoil after there have been setbacks and to recover? And so using the metaphors that the family uses helpful for me as well. The next question is, in light of this understanding, given what you know about your child's illness, what are you hoping for? And David alluded to this as well, that the hope that you might have had for your child before the diagnosis changes when you have the diagnosis, but the hope doesn't go away. We're extraordinarily resilient in our ability to continue to find hope. And I've seen this in families at the very, very end of their children's lives, when there's still a hope for a certain kind of end of their life, or of connection with other people, or even for what they might experience after they die. And so these are important questions. So what are you hoping for? And then the next related question, what are your worries? I asked the question, how do you maintain your strength? And there's a whole complete talk about this, about where we find strength and resilience to manage and address difficult moments in our lives, and also just get through the day-to-day. So I could talk for a long time about that, but in general, it's sort of, what are your internal resources? What are your community resources? And how do you think spiritually or existentially about the difficulties, the challenges that you come up against in your life? And then finally, and this is particularly relevant for families who are in the hospital where there may have been an acute change, how are you doing? How are you coping right now? I'm surprised how often I'll hear from a family, well, we have 3 kids at home, we can't get into family house, we're having to sleep in the car, and going back and forth, and there's just an extraordinary amount of stress in just managing their day-to-day things. And so we can work with social workers to try to make those moments a little bit easier for families as they go through difficult times. Next slide, please. So I wanted to show you this slide, which comes from a colleague and friend of mine, Joan Marston. And it's an interesting slide because it shows internationally what the access is to pediatric palliative care. The darker the color, the more access there is. So you'll see in the United States, in Australia, parts of Europe, and particularly Britain and Germany, there's the darkest colors. That is to say, the most access to pediatric palliative care. But I want to, and you see actually the extraordinary deficits of care in developing countries, especially in the African continent, as well as South America. But if you go to the next slide, you'll see that that actually is a little bit misleading, because when you look at the United States, yes, we have a lot of pediatric palliative care providers, but they're concentrated in big cities where there are large academic centers. So this slide is 10 years old, and so there's some change, but it's not a I wouldn't say that this map would be much darker right now, as there wouldn't be a lot more dots. There are approximately 200 to 300 board certified pediatric palliative care doctors in the country. And so you can imagine with, that's about on average 4 per state, but I know, for example, that we have probably 12 of us are in the Bay Area. And so that's already a fairly significant chunk. So there are large parts of the country that are not covered. I'm not sure why Alaska is not in this picture. It should be. But you see there are no pediatric palliative care providers in Montana or Wyoming or North Dakota. And I think that's still the case even at this time. And so where do those kids go to get their pediatric palliative care? That's a disparity that exists, interestingly. And it just interesting politically too, it's probably correlates a little bit with the rural urban divide that we see that has a lot of political impact as well. So next slide, please. So as one of our jobs as a pediatric palliative care team is to help families think about advanced care discussions. So if a child has a change in status, if there's a worry about a child's condition deteriorating, what are the things that a family would want to do? And that can be everything from focusing on comfort and making sure that a child's day, that moment is as comfortable as possible, even if it means they may not live as long, to the use of medical technology to keep children alive as long as possible, even if that means that they may not be able to be at home or may not be able to be at home in the same way. And those are a wide range of possibilities. They all represent loving choices, and they're all equally accepted from our perspective. But I think the real question is, are we having the conversations about what's important and appropriate for families in a timely way? And most clinicians believe that these discussions are happening too late. Next slide. So our hope as a palliative care team is to cultivate a mutually respectful clinician-family relationship. We want you to feel safe with us. We want you to feel trust in us. And we want you to be able to bring your fears and anxieties. And sometimes those things can be hard to express to your the clinician who's helping to care for your child most directly, especially if there's a clinical trial or something like that involved. You may want to not share some of the things that are worrisome. Some people worry about introducing negative thoughts into the relationship. And we're a place where you can bring those things. We want to be non-judgmental. We want to normalize your thoughts and feelings and use language as I described here as a common thought. And also loving families may make different choices in similar situations. They all reflect deep love for their child, but may also reflect things that are different in importance from family to family. Next slide. So I'm going to transition here and talk a little bit about symptoms that concern parents and caregivers. If I asked you to write these into the chat box, I suspect many of these things would come up. I can't say that I will hit all of them. And I think that if you click one more time, there's another on this slide. I think there's another set of symptoms. In any case, pain, delirium, and agitation, dyspnea, which means shortness of breath or the sense of air hunger, not being able to get enough air, respiratory secretions, coughing or choking on those, and nausea or vomiting. Next slide. And again, fatigue, so, not wanting to eat, which can be extremely troubling for families, having problems with gastrointestinal issues like diarrhea, constipation, and then existential questions for a child who's might feel that they're sick or may be aware that their life is nearing its end, and what are the struggles that they may have with that, and for parents as well. Okay, next slide, please. So I get the wonderful blessing of working with Abby here, who is our therapy dog at Children's Hospital. This particular photo came, my colleague Hania, who is her owner and caretaker and takes to the hospital, said Abby said that there was a vacuum cleaner and it was terrible, and so she needed a hug in order to be consoled. But Abby is one of the most important members of our team, if not the most important member of our team at times, because she just is a representative of pure love and care for children. And she brings a lot of joy and and comfort to kids. And so we bring a lot of folks to the table in trying to care for your child. The Child Life Services, we work closely with them along with music therapy and creative arts. We involve you and other members of the family and avoiding the use of medications as much as possible. And then we have integrative medicine teams at both the hospitals where I work. That includes acupressure primarily, not so much puncture, but acupressure, some biofeedback, massage, hypnosis is also part of what we offer at both hospitals. Next slide. And then, of course, I'm a doctor and trained to use medications to help relieve pain, nausea, shortness of breath, and secretions. As a rehabilitation medicine doctor, I'm also particularly interested in things like ice, heat, and massage, hands-on kinds of therapies, and then procedures, injections, and nerve blocks for kids who are having certain kinds of pain. Next slide. Just want to point out again, these are the interventions that pediatric palliative care consultative services will involve or engage with. So social work, occupational physical therapies, chaplaincy, volunteers, etc. So you see there's a wide range of things that we're thinking about and trying to enlist in the care of the child while they're in the hospital. And so many of these things will be true at home as well. Okay, next slide. So in summary, this is trans or multidisciplinary care for children with serious illness and their families. And by transdisciplinary care, I mean that all of us on the team have some level of expertise in other areas. So for example, our social worker can ask some questions about symptom management. And as a physician, I can ask some questions about spiritual issues that might arise for a family. And so we have some cross-training with our colleagues that helps us be more comprehensive in our assessment. As I said before, there are significant disparities in access to palliative care around the US and the world. And we strive to have exquisite and very careful communication and symptom management at all phases of the life cycle and all phases of from diagnosis through treatment. And so with that, I will conclude and thank you very much for your attention and welcome questions if we have time for that.
Thank you. Yes, so we are running a little bit short on time, but we can take a few questions and really can open it up for questions on any of the topics that we just went through. And I do have, it looks like one that came through, and I'm not sure who's the best person to answer this, but the question is, if you do one of the clinical trials, are you able to do another one in the future if the one you do isn't successful? And I'm guessing that's kind of towards the family clinical trial experiences group. And I guess since I was a part of that, you know, I did ask that question before I accepted our girls into the trial, and everything's going to depend on the clinical trial, you know, that you're applying for. But in most circumstances, you wouldn't be able to be a part of a different clinical trial that is for a treatment unless they allow that. And however, you know, if we get to the point where, say, medication is approved, then you would have access to that even if you've been a part of a clinical trial. I don't know if there's anybody else that wants to jump in on that question, and or if there's any other questions. And all of the speakers are welcome to jump back on camera.
Oh, in terms of that question about the other clinical trials, I would say that gene therapy is a different thing than something that washes out. So like if you were in a clinical trial that has a pill and it has a washout, and that's very different. So whereas I think if you're first in a gene therapy, then it's much harder to I don't think most of the trials would accept someone with gene therapy into like taking a pill. So if you, so that's a major difference.
That's a great point. All right, I'm not seeing any other questions. So Christine, I'll pass it back off to you.
All right, well, I'm gonna keep this brief because we are running short on time, but for the next session with Dr. Al, this is a families only session. And so what we did is we sent a separate Zoom link a couple hours ago. So you would exit out of this Zoom and then find the email or the calendar invite for the next family session. And that one will not be in webinar mode, and we'll all be able to come on camera. And so if you want to join, that is what is going to be happening. With Dr. Al is going to share his story to help the conversation along. I don't have long closing remarks for you. This has been an amazing day. I'm incredibly thankful to everyone for their participation, the sponsors, the speakers, everyone, the board, all the volunteers, Jeff and Caitlin, just everyone. And I do really, I really want people to come to the GM1 externally led patient-focused drug development meeting with FDA. It is such an important opportunity. FDA staff will be watching. There are these live polls, and we need people to do the live polls to say, like, I have GM1 type 1, type 2A, type 2B, like I'm from this part of the world. Like, it's going to be really interesting. And I just, please, if you can, even if you can only go to a certain part of it, or if you're going to miss it entirely, there will be recordings and the opportunity just to type in comments. So please, please come and please join us for Dr. Al's session. So exit out of this and find the other invite and go to that Zoom link. But thank you everyone for an amazing day. I appreciate it so much. Thank you.